Hello, 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 everybody. Welcome to the stream. Hello. How are you all doing? It's been a little bit of time since I've seen all of you, that I've gotten the chance to talk to all of you. It has been a pretty hectic last couple of days, considering we are now approaching the end of the semester. I only have one more assignment to grade before I am transitioning into a full-time content creator, which is both exciting and of course terrifying. Yeah, I'll start off by saying hello to all of you. Hello. Welcome Somewhat and Seon. Welcome. Welcome Human Buddy and Robert. Good luck on your interview. Hello, Acedium, welcome, and Astral, Archie, welcome, Lionheart, hello. Yeah, welcome. So, I was originally not going to be streaming today, considering, yeah, just we are winding down the semester and I have a bunch of grading to do, but I really wanted to chat, and so I wanted to find out if there was a way that I could make some content without it being too stressful. And I think it was from the Discord that had pointed out that that there were papers that had answered or were trying to get around this answer of how are prions transmitted to the brain. And this was all brought up because of a new YouTuber, quote unquote new YouTuber on the scene. And we'll talk a little bit about exactly what inspired this stream in just a second now. So there's a YouTuber called Smoreside with a very familiar voice, for those of you who know, if you know, who made this video called The Chronic Wasting Disease Iceberg, which was very good video. It's a video that does not actually overlap very much at all with my own prion video, because I focused a lot more on molecular mechanisms. This is more about the spread, the epidemiology of this disease. And what was noteworthy to me is that according to this video, there have been apparently, I don't want to get the number totally wrong, so please don't take this for a literal recollection of the video, but from what I recall, from what I saw, that there have been about 1 million people in the UK exposed to the mad cow protein. And if you've watched any of my prion content before, you would know that, you know, prion diseases are some of the worst diseases known to mankind. They are, of course, very rare, so they're not that scary in that regard. Oh, is the Discord link not working? That is unusual. Let's see. Yeah, I'll see what I can do about that. Yeah, maybe towards the end, or maybe I'll link it as a comment of this video so you can check out the VOD later if you have to go. Yeah, unfortunate. I do have the setup so that the Discord link should be permanent, but I'll have to yeah, check back on this stream later, and I'll just add it in the comment section. But yeah, like 1 million people exposed to mad cow protein, which, you know, if you had told me that as a kid, I would be absolutely terrified. But we know that this is just exposures, and we know now that, you know, we don't have 1 million people in the UK dead to mad cow disease, and it has been 30 years. So the story has to be a little more complicated than that. And I think that is what I want to talk about today. The fact that even though there has been significant exposure to mad cow protein in the UK, there's still some sort of barrier to an active prion infection. And that's something that this wonderful video brought to light for me. And it's something that I probably should have covered in my own video, but it just goes to show how difficult it is to cover 100% of any given topic, right? Like, I thought for many months that, you know, my prion video was quite complete, but here is a huge blind spot that I never got to address, right? So let's talk about that. So if millions of Brits were potentially exposed to mad cow disease, how come we're not seeing millions of Brits with prion diseases, right? Prion disease remains to be an extremely, extremely rare disease. I'm talking about under 400 cases worldwide, ever, you know, in terms of mad cow, not in terms of prion diseases in general, but in terms of transmissible prion diseases that you get from eating a cow with prion disease. And so there are a couple of different hypotheses as to why we have not seen such a terrible outcome from the mad cow, I don't know what you would call it, breakout incident that occurred in the early 90s. And so one hypothesis is that it just hasn't been long enough, but it's been, you know, about 30 years. That's a pretty long time for a disease to be incubating. And then we have alternate hypotheses like limited disease penetrance, like maybe, you know, maybe the prion protein itself, the misfolded prion protein, just has a variable ability to penetrate into human tissue somehow. Maybe there are individual mutations within the prion protein or different folding forms of the prion protein that make it harder for some individuals to be infected than others. Or maybe there's some sort of genetic resistance on the part of the host that enables there to be a tougher barrier to infection. In any case, what is true is that given the number of people that have been potentially exposed to the mad cow prion, we see surprisingly few people afflicted by this disease, which is very, very interesting. Hey, Gluon, welcome. And Dr. Weebmats, worse yet, tools infected by prions can't be cleared by normal means because prions are so hardy. Yeah, I did go over that in my prion video where, you know, even autoclaving has trouble getting rid of prion disease. Maybe it depends on the dose of prions. I'm sure that dosage is something that is important. Generally, if you have more of a bad thing, it means more bad. However, the fact that there are people that have died very quickly from this disease and people that maybe have yet to die from this disease 30 years later suggests that dosage might not be the top effect determining this. So let's get into some of the preliminary research trying to identify whether or not we're about to have like a mad cow apocalypse. So what was noted about the initial mad cow outbreak is that patients with homozygous, oh my gosh, oh my gosh, I can't speak today. Patients with oh my goodness gracious. I cannot speak today. Welcome, Ethan. Welcome. People who have 2 genetic copies of the methionine variant at position 129 of the prion protein, as you can see highlighted by this arrow here, this amino acid here, if this is methionine, these patients were a lot more susceptible to mad cow disease. In fact, these are basically all of the patients that died immediately following the mad cow outbreak. So if you had 2 copies of methionine, so both of your chromosomes, both copies of that particular locus, if that had the methionine variant, you were unfortunately very, very vulnerable to prion disease. In fact, yeah, the entire first wave of this disease happened because of this homozygosity. My goodness. Maybe the immune system can consume it somehow. I mean, there should be something there since it exists in nature. We'll look at that, Sierra Smith. That's a very interesting thought that we will address. Welcome, True Octagon. Welcome. How do you know that? So I have links to the review paper in the description, I believe. If not, then I will have it up shortly. But this is all from like review papers and press releases about the potential second wave of prion protein. So this is an article from New Science that claims that 1 in 2,000 appendices from appendectomies in the UK have the presence of the mad cow prion. But even accounting for the prevalence of these homozygous methionine carriers, there are fewer cases of active prion infection than expected, although that could change. Although it also has been a very, very long time, right? It's been 10 years, roughly, since this article was published. So it's pretty nuts, honestly. Eventually there would be natural selection against this variant, depending, because it is such a long incubating disease, it is kind of hard to select against this kind of thing. Well, at least, through the mechanisms of natural selection, at least. Can protein unfolding enzymes be modified to attack it? We will talk about whether or not unfolding and enzymatic degradation is actually the strategy, because it's complicated, although it is very interesting. But yeah, so this is actually like really, really crazy here, right? The fact that 1 in 2,000 people who needed appendectomies have been identified to contain the mad cow prion in the UK. And yet we do not see 1 in 2,000 people in the UK succumb to mad cow disease. So there has to be something more than just the homozygosity, which exists in about 30% of people who live in the UK. And so there has to be something else about biology that is preventing people from having it as bad as the people who had it in the first wave. So I hope that answers why we're talking about the appendix. It's the fact that even though so many Britons were exposed to the mad cow disease, we see, not even 200 cases of mad cow disease actually appear in the UK, which means there's something more complicated going on. And the fact that exposure to mad cow prion protein was so prevalent is not translating into actual disease is a very interesting disconnect that I think we need to examine. Should bodies of cows or other things that have the prion detected be fully incinerated? So that is what the UK government did in 1990, which was, tragic, obviously. Like I'll say this in the most neutral way possible, right? Like a lot of our commerce, a lot of our agriculture is dependent on herd animals. A lot of our current food supply chain is dependent on the meat of these animals. And because this was and currently is such a huge problem in terms of zoonotic infectious agents, a lot of cows were slaughtered and incinerated, which ultimately I think for the well-being of humanity ended up being a good thing. Although good is not 100% one way and 0% the other, right? It's shades of gray here. Yeah. All right, so there is this disconnect with exposure to prion protein and actual disease. So what's up with that? So that brings up a couple of interesting hypotheses, right? So again, the prevalence of carrying the mad cow prion in the UK is surprisingly high, with 1 in 2,000 people who needed an appendectomy. So I'm being very careful to state it this way because this does not mean 1 in 2,000 people in the UK have are carriers for the mad cow prion. This is just 1 out of 2,000 appendixes that were examined for prion protein, right? So it is noteworthy, but you can't really generalize too much to the entire population, but it is still surprisingly high given the brief period of time where mad cow became a thing, right? So over the course of 26 years, only 178 cases of the disease, not carrying it, but the disease itself have been reported in the UK, right? So that's a very low number of cases given how prevalent beef consumption actually is. And the homozygous MET variant of the prion protein in the host is carried by about 30% of individuals in the UK, and so that is quite a bit of people that would be susceptible to this disease. And again, we are not seeing total apocalyptic fallout of this disease yet. Could be that we just haven't had the time, that it's really, really slow in individuals for some reason, or it's because there's some other barrier that is preventing this disease from happening in the majority of people exposed. All right. Is that 178 only for humans? It's only for humans. Every once in a while, you'll get a cow with prion disease, and they are usually removed immediately. And every time it happens, it is like newsworthy, but it's usually in individual cases. Like maybe that particular cow had a spontaneous mutation that caused the prion disease, which of course can happen. That cow is removed and incinerated to prevent spread. Hello, greetings from Cthulhu. Thank you so much, Cthulhu, for the super chat. Thank you. I hope you enjoy the contents of the rest of the stream. Yeah, so I find this to be really interesting because I was not really aware that the level of exposure, potential exposure, was that high, and that so many people in the UK are carrying the prion protein without actual prion disease, which for those of you who are like me that have had like intense prion fear, this is actually really, really hopeful, right? Like I never want to have my channel be clickbait for fear, right? That's not the point of my channel, right? I always end my videos with, discussions of medication, vaccines, treatments, etc. And this is something that I was so excited to share because, this is indicative that, like, even though prions are omega deadly, some of the worst diseases known to mankind, we don't fully know how this disease occurs, and maybe finding out more will actually allow us to breathe a little bit, right? Because even in my video, I was like, oh, if prions continue to aggregate in the environment, we could see a situation where we have an apocalyptic level of replicating prions, and then all of our food is bad, and then we all die, right? That's the apocalypse situation. But this data kind of suggests that something is happening where the disease is not actually that penetrant, and I want to examine a couple of things that I learned in the last 2 hours, actually, because this was the most last minute I've ever done one of these before, to kind of give us some hope so that we're not filled with this kind of anxiety forever, right? Yeah. Are these prions only affecting mammals, or can it jump to non-mammals like avians, reptiles, or fish? So avians have their own prion protein. I would be really surprised if mammalian prion protein could affect an avian prion protein. Reptiles, I don't think lizards have their own mammalian prion protein, right? They're not mammals. Fish have their own super old version of the prion protein, but is not like it's only got like 30% conservation with mammals, specifically in the zebrafish. So my answer to that would be I wouldn't worry about other mammals, but I would be worried about plants potentially being a potential vector, because research has shown that plants can actually uptake the prion protein. Now, whether or not that means that we are going to be exposed via plants is a different story, because again, I want people to understand that the thesis of this stream right now is that the apocalypse situation has not happened. Life has been around for so many years. You know, mammals have been around for so many years. Prion diseases have been around for so many years. We're all still here alive today, which means that there's probably something preventing a prion apocalypse from happening. And I want to just remind everybody that, because I feel from the chat that we're getting into fear territory, and I want to keep us out of the fear territory. All right, we're not here to fear today. This feeds my hyperfixation. I mean, join the party, right? I've been obsessed with prions since I was like 12. I thought prions were a general thing. They are a general class of proteins, but the prion that is responsible for disease in humans is the mammalian prion protein. I mean, you could argue amyloid beta, you could argue, tau, alpha-synuclein, whatever you want. But in terms of when I say prion disease, I mean literally the prion protein, the thing that is called the prion protein. Because while amyloid beta might be classified as a type of prion, there is only one prion protein whose name is prion protein. I hope that's a bit clearer. Okay, so maybe for most people, prion never reaches the brain, right? We already know from these appendectomies that it's surprisingly easy for the prion protein, the misfolded, mad cow prion protein, to end up in the appendix, but it seems a lot harder to reach the brain. So the big question for today is, what's stopping the prion from getting to the brain? How does the prion get into the brain at all, right? Like, that is going to be really essential for determining what our actual risk is. And, I'm not an epidemiologist, and this is not my field of major specialty, so I couldn't definitively tell you what's going on here. But this is, again, I'm reporting on some things that I've learned in the last 2 hours. All right. What do I mean by the misfolded version? So you have 2 copies of the mammalian prion protein expressed primarily in your neurons and sometimes in the skin and the gut. The prion protein itself is a very normal part of your biology, and when it's normally folded, it's probably doing something like helping your neurons talk to each other. When you get a prion disease, that occurs when the prion protein misfolds, causing aggregation, which can be cytotoxic, killing your cells. And so if you want to learn more about the basic stuff about prion disease, you can click on my channel and go to my videos tab. It's one of my most popular videos, should be very easy to find. That'll bring you up to speed on how prions function in general. Thank you for subscribing, H2O Addict. All right. So let us continue here. So here is the paper that we will be reading today. This one, I am going to speed run the reading. I'm actually going to read it the way I would read it if I weren't reading it for content, which means skipping around a lot. So I'll show you how I read a paper in particular, right? So this is not let's read together. This is I am showing you how I would read a paper and kind of translating for you as I go. The reason why I want to do this is because this is actually not the focus of today, right? So the focus of today is going to be a review paper that actually answers my question. This is actually just a paper that is a piece of that overall story. So if we're thinking about, how does the prion reach the brain? Something that you always have to think about with regards to things reaching the brain is the blood-brain barrier, right? So not for the spreadsheet. Yeah, Astral, I think this would be cheating if you were to put this in the spreadsheet, because I'm not going to be reading words. I'm just going to be looking at data, essentially. So this is going to be me looking at the figures, because I want to spend more time talking about this review paper that I can't read on stream because it's copyright paywalled, which is super unfortunate. So I will switch now from the slides, and I will actually switch to the PDF that I have. Bye, Robert. Good luck on your interview. Let's load up that PDF. So I've got the PDF up. All right? So again, just to give proper credit, this is a paper from PLOS Biology, open access. We love open access, true heroes here. And their title is Prion Pathogenesis is Unaltered in a Mouse Strain with a Permeable Blood-Brain Barrier, right? So we know that a huge barrier to delivering anything to the brain is the blood-brain barrier. There is a solid quote-unquote wall that prevents things like drugs and bacteria and stuff from getting through, although it's not totally impenetrable, right? It stops a lot, but it is not perfect. And so a reasonable hypothesis is that maybe the prion protein, the misfolded prion protein that causes disease, is something that travels through the blood and reaches the brain that way. And so we're just going to go ahead and read the abstract here. I'll read the abstract out loud, and then I'm going to just look at the figures actually. So here's the abstract. Transmissible spongiform encephalopathies, so these are prion diseases, are caused by the prion, which consists essentially of PRPSC. So this is prion protein scrappy. This is what people in the field refer to the disease version of the prion protein as, scrappy being a prion disease found in sheep. So they named the misfolded protein after the sheep disease. An aggregated conformationally modified form of the cellular prion protein, PRPC, so this is normal healthy conformation. Although these prion diseases can be experimentally transmitted by intracerebral inoculation, so you can give an animal a prion disease by inoculating their brain, that's not surprising perhaps. Most instances of infection in the field occur through extracerebral routes. I would say that all infection in the field occur through extracerebral routes, but I get that they're hedging there. The epidemics of Kuru and variant Creutzfeldt-Jakob disease were caused by dietary exposure to prions and parenteral administration of prion-contaminated hormones. Interesting, I didn't actually know that, has caused hundreds of iatrogenic prion disease. I actually don't know what this word means, but I'm not going to worry too much about it. In all these instances, the development of post-exposure prophylaxis relies on understanding how prions propagate from the site of entry to the brain. While much evidence points to lympho-reticular invasion followed by retrograde transfer through peripheral nerves, prions are present in the blood and may conceivably cross the blood-brain barrier directly. Here we have addressed the role of the blood-brain barrier, BBB, in prion disease propagation using these knockout mice, which possess a highly permeable blood-brain barrier. We've found that these mutant mice have a similar prion disease incubation as their littermate cousins, or littermate controls, regardless of the route of prion transmission. These surprising results indicate that the blood-brain barrier permeability is irrelevant to the initiation of prion disease, even when prions are administered parenterally. I don't know what that word means either. Iatrogenic, caused by medical intervention themselves. Is that what that means? If so, I learned a new word today. Very interesting. I've read a lot about prion diseases, and so me reading about the background doesn't really do much for me. I did read the methods beforehand, which I'm not going to get into because it makes for pretty uninteresting content. But essentially, this is what I wanted to get to. So you have these mice, half of them are wild type, aka healthy and normal, and then the other type have a mutation which causes their blood-brain barrier to be kind of leaky. And so these mice were infected with the Rocky Mountain Laboratory prion strain, and they were injected intracerebrally, so directly into the skull, intravenous, so through the blood circulation, and intraperitoneally, so this is within the body cavity, right? So just injected through not the stomach, the organ, but like the body cavity that houses the stomach. Parenteral is something to do with intestines, I guess. I think that makes sense to me. Prenatal, I don't think that parenteral is the same as prenatal, but we will be able to deduce the context, I think, later. This is what I get for not reading the paper beforehand. Scrappy is the sheep disease. Yes, it is. So the PRPSC is the misfolded protein named after the sheep disease. Iatrogenesis is the causation of disease, a harmful complication or other ill effect by any medical activity. I see, yes. So there were outbreaks of the prion disease that have occurred because medical supplies were contaminated with misfolded prion. It means injected. All right, thank you. Thank you, Mandrake. Okay. Okay, so one group of mice was inoculated intracerebrally with brain homogenate containing this amount of infectious agent. That sounds nasty. And then a bunch of things were done in order to assess whether or not you had a meaningful prion disease. Let's just put it that way. So let's just go ahead and take a look at the figures. So I will expand the OBS thing because it looks like we're not quite there. Great. That looks like we have all of that on the slide. So it says figure 1, increased BBB permeability does not alter prion disease incubation. So here we have survival curves, right? And we have differing amounts of the initial prion load, and we have a couple of different conditions, right? So here in blue, we have the wild type mouse. This is the healthy mouse. The red is the heterozygous variant. So this mouse has one copy of the healthy blood-brain barrier gene and then one copy of the leaky blood-brain barrier gene. And then in black, we have the mouse that is homozygous, 2 copies of the leaky blood-brain barrier. And when we look at the survival curves, days post-inoculation, we see that they all kind of survive just about the same amount of time. In fact, we see up here, in the 1.5 log LD50, we actually see that for some reason the leaky blood-brain barrier mice survive the longest. But if you take a look at the trends over these different conditions, you'll see that there's not really a clear winner out here. They all look similar, so I assume the disease is unchanged. Yeah, so it looks like having a leakier blood-brain barrier doesn't actually change how quickly you die from a prion disease, at least when we're working with mice and injecting, right? So the other noteworthy thing to know is that these mice are not exposed to the prion protein in a, we'll call it physiologically relevant way, right? One group was injected in the brain, one of them was injected through the veins, which is again not really the way that regular prion diseases happen, and the other was injected in the body cavity, so not on the inside of the GI tract, but the outside of the GI tract. So none of these are, frustratingly, none of these are physiologically, what we would expect to happen in the wild. So that doesn't mean that this paper is useless. It just means that we have to remember that there is an aspect of this that does not reflect reality. Hello, Artistic. Welcome. Okay, so that's surprising. And now here we get some histopathology. So what do we have here? Figure 2, prion histopathology and evacuation after the brain homogenate inoculation. So this is looking at, I think, vacuoles per FOV. I think this is looking for holes, right? So I believe, I can just look this up really quickly. Vacuoles. Oh yeah, this is just regular vacuole. So I think they're looking for gaps in the brains of the mice. So this is indicative of a brain damage, and trying to see through histopathology whether or not you have more damaged brain matter if you have a leaky blood-brain barrier. So if we just take a look at the data, we'll say, so this is IP up here. So this is intraperitoneally. Oh God, I'm so bad at saying these words. But these were mice that were injected through the body cavity. We have wild type. We have one copy of the bad blood-brain barrier, and then we have 2 copies of the bad blood-brain barrier. And we're looking at the frequency of these vacuoles, so these holes, and we're checking for, per field of view, how many of these damaged holes we see in the brain tissue. Prions are just confusing. They are, after having done the flu virus, I think prions are not exactly confusing, but rather just not a lot is known about them. I think that's because per dollar spent, you're not getting that big of a return, right? In terms of medical burden, diseases that stem from the major mammalian prion protein don't actually make up that much of the human population, and so there aren't a lot there's not a lot of interest versus cancer or something like that, right? Not to say that there isn't any interest, right? The prion protein is still pretty well studied. It's just not as well studied as stuff that's involved in cancer and stuff that affects more people. It's just, a matter of funding. Yeah. Am I late? Did I miss a lot of stuff? So I'll recap sort of what motivated me real quick to do this journal club today, this half journal club, half paper reading, is that there have been a surprising number of exposures in the UK to mad cow prion. And in terms of finding the prion protein, the deadly prion protein in people, a lot of people actually are carriers for the prion protein, but not a lot of people actually show the disease. And that's not to make people afraid. That's to ask the question, what about these people that are carriers of the prion protein makes them resistant to actually developing a disease? With the caveat that it has only, quote unquote, only been 30 years since the mad cow outbreak. So it could be that in another 30 years, we'll start seeing a disastrous second wave. But the fact that it's been 30 years and nothing has really happened, that makes me think that something else is going on. I have a question. If prions are just proteins, why can't the cell just store them up, like in vacuoles or in granules? It's an excellent question, and that would require there to be some evolutionary pressure to ensure that that particular protein is tucked away. And because prion diseases are so rare, I think that there's not been a lot of selective pressure to evolving something like that, right? So we'll actually look at mechanisms of cell-cell transmission, but I think from a macro point of view, right, a lot of answers in biology are just, well, there wasn't enough selective pressure. Carriers of prion disease, new fear unlocked, don't worry. Again, this is not the fear stream, okay? This is taking a look at the very hopeful real data that of the many people potentially exposed to prion diseases worldwide, there have only been about 400 cases of mad cow disease, and it's been 30 years. And to me, that is very, very hopeful. Very, very hopeful. And so if we look down at this histopathology and we're counting the number of vacuoles formed in the prion infected brain matter of these mice, I do kind of agree that if you were to tell me, like if you were to randomly blind me, I couldn't really tell which one is wild type and which one is blood-brain barrier leaky. Cells haven't put points into that skill yet. Yeah, exactly. And I actually don't really think they ever will, considering how rare this stuff is. And as we'll see after I finish this paper, there might be other biology things that are preventing prions from ever making it to the brain. And I want to get into that. Is this a classroom or a fun hobby? It's interesting either way. So this is actually going to be my full-time job in literally 1 week. So I am a biology instructor at the university level. I was actually told that my contract would not be renewing. So in 1 week, YouTube will actually be my full-time job. And I'm very excited for that to be the case. So it's kind of halfway between a fun hobby and a classroom right now, right? Yeah. If you're new to my channel, which I'm really surprised by, because I think most people have probably watched my videos before my stream, but if you're new here to who I am, I'm Phi the Neutrophil. I make videos about cell and molecular biology, and I love talking with people about science, even though like my videos do way better than my streams. It's still a way for me to scratch that part of my brain that loves talking to people about the science that I love so much. Rip, sorry about that. Exciting though? It is exciting, and I would probably be trying to find an actual real other job if it weren't for, well, I'll still apply because why not, right? But I would never think about doing it full time if I didn't have the support that I have. So I'm very grateful to all of you. Shout out to members too. of course. Yeah, where was I? Yeah, so this disconnect of like, why have we not seen a mad cow apocalypse? I think there's a lot going on that we don't know about that might explain our relative resistance to this kind of disease. Prion fibrils look like modern art, the cryo-EM. Yeah, exactly, like from the thumbnail. And, Clockwork is the perfect person to appreciate structural beauty with. Okay, so what is this figure? scrappy deposition after inoculation in control and blood-brain barriers. So these are cerebral cortices. So these are slices of mice brains that have been injected with a prion-containing brain matter, and the darkness, the brownness of the brain tissue will correspond to how much of that prion protein is present. Let's see. So we are trying to see here whether or not we can see whether the leakier blood-brain barrier is darker than the not leaky brain. And we can actually see that if we compare the left column and the right column, they're about the same. In fact, here, down here, right, it even looks like the leaky blood-brain barrier brain slice is actually maybe even a lighter color than the regular healthy mouse brain. So this is coming back over and over again to the same conclusion, that leaky blood-brain barrier, as long as this model of blood-brain barrier leakiness is somewhat physiologically relevant, is indicating that this is maybe not the route that the prions take to the brain, which brings up a very important question. If it's not the blood-brain barrier, right, if it's not going through that route of circulation, how the heck is it making it to the brain? And that is why I wanted to actually do this very, very, very last minute journal club, where I am going to talk through what I learned from this review paper. So again, caveat, this is, I started thinking about doing this journal club literally 2 hours before I hit go live. So this is very rough. It's a very rough summary. The storytelling might not be as good as my, normal style, but I still felt it was fun enough to talk about where I wanted to just do it for all of you here. All right. So we have this review paper. This is the one that I'm going to be talking about today. This is transmission of prions within the gut and towards the central nervous system. This is a review paper published in 2011 by Lands Bioscience that has been acquired more recently by Springer. And if you go and you look at the the copyright, and if you, because I would really love to read these articles out loud to you, but just to give you an idea of how insane publishing is, if I wanted to show this article on stream, it would be $3,000 for a regular normal guy like me to reproduce, which is absolutely insane. Thank you, Eason, for subscribing, or for being a member. Welcome to the progenitor cells. Thank you so much. If it isn't passing through the blood-brain barrier, is it being made in the brain? So the regular healthy version of the prion protein is already being made in the brain. The problem with mad cow is that you eat the mad cow prion protein and that somehow makes it to the brain. So there's no reason to think that eating the mad cow prion protein would cause your brain to make mad cow prion protein. So there should be a route, a delivery route between the gut and the brain, and that's what we'll get into here. Yeah, that's pricey. Yeah, I know. It really, really stinks because I really want to be like very okay about YouTube and fair use and stuff. And the general rule of thumb is that if you don't have to argue your case for fair use, it's best to just not. Even though I think if I were to cover this, it would technically be fair use. I feel very strongly that is the case, but it's not worth risking my livelihood over, essentially. Prions, I think, are entering from the muscles. They get sucked in and just spread. That's how it happens in rabies, right? So you get bitten by a rabid animal, the rabies virus enters the muscle, and then the muscle has a junction with the neuron, and then it spreads to the brain that way. That's not actually going to be the case for prions. Not to say that you're wrong, it's just that that's not one of the hypotheses that's in this paper. Although it's not a bad one, again, because that is how rabies does it. So it's not a bad thought. Pretty interesting. Wait, the brain would make prion protein? Yeah, your brain makes regular healthy prion protein. Regular health. Again, we're not fearing today. If you want to learn more, I do have that prion video on my channel. It's a banger. But yes, your brain makes healthy prion protein. When you get a prion disease, it's because a misfolded, diseased version of that prion protein is causing the healthy version of that protein to misfold in your brain. That's how those diseases spread. Okay, so what does this review paper suggest? So, an interesting finding that this paper reveals is that mice with the correctly folded membrane anchored healthy prion expressed in the gut enabled prion misfolding in the brain. So this doesn't really lead anywhere for me, but this does indicate that, like, if you stopped your gut cells from making regular prion protein, that would actually prevent that prion protein from getting to the brain. So this implies that the misfolded mad cow prion protein probably needs the healthy version of that prion protein to start its trip. I don't really know how, or I don't really know what happens or why it needs that healthy prion protein. My guess is that in order to get the amount of misfolded prion protein that you need to cause disease in the brain, you might need a little bit of kindling, right? So if your gut cells are making the healthy prion protein and that prion protein is being misfolded by mad cow prion, that might give you enough inoculate to make enough misfolded prion to actually be a problem. Although that is not guaranteed. Like, you could think of other mechanisms by which this is true. Maybe the misfolded prion protein needs to bind the healthy prion protein in order to be sucked into the cell. Something like that could be true. I didn't have enough time to fully investigate that line of thought, but it was just an interesting note, I think. All right. So what we actually do know about the potential routes by which the prion protein can make to the brain is through the intestine. So it looks like that there are a couple of different candidates for routing the prion protein from the gut to the brain. And these are the cells of the intestine. So we have a couple of different candidates. One are the enterocytes. One of them are the microfold cells, and one of them are the dendritic cells. And so these three cells are some of the major components of the intestinal lining. And if you get entry from the lumen side, right, you have the prion protein here. These three cells can transmit or help that prion protein make it from the inner intestinal lining to the lymph. So for those of you who don't know, the lymphoid system in your body is like the cellular highway, essentially, is that a lot of things are transmitted through the lymph system, especially stuff that are immune relevant, right? So you have your regular circulatory system that carries blood and stuff, and then you have the lymphatic system, which is a different fluid superhighway throughout the body. So it looks like, this is consistent with this idea that, like, this is probably the route and not something that has to do with the blood-brain barrier, that entering through the lymph might be a way for them to circumvent something like the blood-brain barrier. Like a nucleus or a crystal seed, I wouldn't go that far, because we're about to talk about seeds in a seed specific context. I would just say no. Actually, you're right. You're right. Wild Mags. You can think of it like nucleation. Like, if you didn't have the healthy prion protein, maybe you never start crystallizing. It's not really crystallization, but it's analogous. I think that's not a bad way to think about it. Yeah. So these are candidate cells that might enable the misfolded prion protein to transmit from the inner lining of the gut to the cellular superhighway. We also know that Peyer's patches are required. So here's where I want to shout out anatomists, right? Because I'm like a cell guy, I'm not like an anatomy guy, and so I'm just gonna take them for their word here, where if we have this picture of the intestinal lining, you can imagine the prion protein starting up here. I actually have a pencil tool that I can use. Yeah, like the prion protein being up here and then going through here. And we actually know that Peyer's patches, which are cells that are directly under the intestinal lining, we know that if you remove those Peyer patches, that you stop transmission to the brain. So the cells that lie directly underneath this lining are probably pretty important for sneaking this prion protein directly to the brain. Which, for me is very interesting, because this suggests that immune proteins and immune cells themselves are involved in sneaking this protein through. Okay, so a different review paper, and this is really, really interesting. Wait, does the dendritic cell produce prions? No, so the dendritic cells can take up prions and, send them down the lymphatic system. So we'll read this abstract. Again, this is another review paper that is paywalled, another $3,000 that I would have needed. This is from the abstract, which is publicly available. So I'll read this part from the abstract. Once the intestinal epithelium, migratory bone marrow-derived dendritic cells, DCs, are ideally situated to acquire and transport prions to lymphoid tissue. So answering Artistic's question, the precise involvement of the dendritic cells in pathogenesis is uncertain, but a subpopulation of DCs can acquire disease-specific prion protein from the intestine and transport it to mesenteric lymph nodes. However, and this is really, really exciting, macrophages also acquire prions, but these cells are unlikely to be involved in transport as they destroy them. And I did not know that. I did not know that macrophages are actually capable of destroying these prions. So this is why this is not the fear stream, right? Like, again, the setup is scary, right? The idea that you've been exposed, potentially exposed to a deadly disease is very scary. But as we learn more and as time passes, we're actually finding that there are intrinsic mechanisms in the body that might explain why we're all not all dead yet, right? Like, yeah, like macrophages are OP. How do they destroy them? I don't know for sure. Again, 2 hours since, I started preparing for this stream. But if I had to guess, it might be because macrophages have some of the most insane digestive systems of any cell, which is not necessarily like always a positive. So this is a good time for me to explain why protease engineering is not always the strategy to go when thinking about treating a prion disease, right? Because I forget who talked about seeding before, but it has been shown that treatments that try and chop up the prion protein, sometimes that can lead to worsening outcomes. Sometimes, right? I'm not saying this strategy will never work, but if you do not completely destroy the crap out of the prion protein, what you might end up doing is causing there to be more seeds to form. So you can imagine like destroying the misfolded prion protein, but that, if you destroy one strand, that becomes 8 strands, and now you have 8 potential nucleation points that force other healthy prion proteins to misfold, which can be, as you can imagine, a freaking disaster. So the fact that macrophages can thoroughly destroy prion protein, not just cut, but chew them up, is really, really encouraging, and again might explain why we have not seen apocalyptic levels of prion protein diseases accrue just in general, right? So even though prion proteins can accumulate in the environment, even though plants can uptake these prion proteins, even though this and that, the body might have defenses that prevent you from automatically dying to this very high penetrance, or not high penetrance, but high lethality disease, right? If you look at the Wikipedia's most deadly diseases, prions are number 1. If you have the symptoms, you die 100% of the time, essentially. But that is before taking into account how many people are actually carriers, right? So knowledge is imperfect, and part of why I love like doing this channel so much is that I get to learn a lot of different areas of biology. Like I get to synthesize from all sorts of different fields, and so I kind of had felt like there was a piece missing, but I couldn't really identify what that piece was. And I think having this part of the prion story is really satisfying, right? So I'm not saying that like I don't want to be too hopeful, right? Because there is that kind of idea that maybe it just hasn't been a long time. But what if it takes 60 years for, someone without the methionine homozygous variant to have prion disease? Well, that means you still get 60 years of good life. And so maybe a key player in stopping that from happening is the macrophage, although the extent to which macrophages prevent prion disease is not really well known yet, but yeah, so I do think that it is very, very interesting, like a hydra. Exactly. So I think protease engineering for prion diseases, it's a nice idea, but it has to be done extremely thoroughly because you do not want to make seeds, essentially. I made a model of Ebola's destruction of the immune system as a science fair project in the 10th grade using your video. Thank you so much. That's awesome. If you want to like post it anywhere. Again, I'll link the Discord at the end of stream if the Discord link isn't working, and just like remove anything doxable. That would be really cool. You don't have to, of course, but it would be neat. Do we have that many macrophages to take care of how many prions that are floating around in such a large area? Yeah, that's an excellent question, and I do not know. And it's something that I think that people will eventually find out. This is, of course, a paper from 2006, so it'd be a very interesting follow-up here. Do we know what types of macrophages can destroy prions? Like, can alveolar macrophages do that? I would guess that alveolar macrophages are not going to be able to do it just because they're not really specialized to do protein stuff. But I don't know. It's an interesting question. Okay, so summary so far. Actually, maybe I'll just link the... No, it should be working, it looks like, right? We just had someone join. Yeah, it should be okay. Should be okay. So yes, that link should be good then. Okay, so here's a summary so far, just in case you are coming in. Macrophages in the brain. So those are called microglia. So there are macrophages in the brain, but they're called something different. So the blood-brain barrier, probably unimportant, although don't know that for sure. Looks unimportant in terms of the mouse model. Misfolded prion requires normal prion in the gut to get to spread. Don't really know what the mechanism by which that occurs is, but it is an interesting note. Prion is first trafficked to the lymphatic system by cells in the intestine. That's somewhat unsurprising. But then how do we get to neurons, right? So this is the next question that I had. So we have the connection between gut and the lymph, which I think is very interesting. What about the neurons? I wonder if prion rides around in dendritic cells like Ebola. I mean, I didn't get into that much detail in the time that I had, but, you know, it'd certainly be interesting to follow up with that. So from the lymphatic system, there are actually a couple of hypothesized routes. So the splanchnic nerve, the lumbar spinal cord / vagus nerve. So these two are the leading potential candidates for how the prion protein gets from the lymph to the brain. And again, I am not an anatomist, but I think acknowledging one's own limitations and acknowledging the specialty of others, I think it taps something very deep into my brain that is like very shonen protagonist in a kind of way, right? Like, you know, any ensemble shonen anime like My Hero or Naruto, I always feel like there's a part of me that has this immense respect for someone else with a different skill set than I do. And so while I have been very happy to learn about the molecular mechanism by which prions misfold and stuff, and sort of the cellular aspects of the prion protein, you can tackle the prion problem from all sorts of different levels, right? So the level that I like to tackle is like level 2, right? I think level 1 is basic physics of the prion protein. Like, how does the prion misfold physically? You know, what are the bonds involved? What are the physics involved? What is the thermodynamics involved? And then there's like level 2, which is how does the molecule act? How does the cell act? And then this, I would say, is like level 4 or level 5, where we're talking about whole systems and the body as a whole. So this is like the realm of anatomists and doctors and stuff who have to look at the body as a whole. And if you are thinking about med school, you will have to learn like every bone in the body and every nerve and everything. I don't have the memory for that kind of stuff, but it does show you how valuable it is to have different people with different skill sets, because I would not be able to tell you what any of this actually means. But it is interesting that we kind of have an idea of what is possibly happening, that we have transmission from the gut through the lymphatic system that gets sneaked through, you know, 1 or 2 different main neurons that hook up to the central nervous system. The power of friendship. Yeah, exactly. Like, you know, back when I was at grad school, when I was listening to friends of mine present their work, I always had such admiration, and I thought they were really cool. And I think if I were, you know, a little ninja child in the Leaf Village, I would feel that like same admiration for the hyper, you know, specific skill sets that people refine and develop. I think it's very, very cool. And, you know, as a selling point for science or for pursuing higher education, is that you get to be that like super specialized, hyper knowledgeable person. And I think that's really dope. I mean, of course, you know, getting a PhD is not for everybody, but choosing to specialize doesn't have to be a PhD. It could be any level of like rigorous specialization. Doesn't even have to be in the academy, right? Any level of I dedicated my whole life to doing this one thing is really admirable and awesome to see, I think. Yeah, so shout out to Anatomus here. Really, really incredible, deep thinking about how the body is routed, and I respect that. It's not for me, but I respect it. So summary so far, and Joel, this is a good place for you to jump in because I'm going to summarize what we've learned so far about transmission of the prion protein. The blood-brain barrier, probably not important. Misfolded prion requires the normal prion in the gut to spread, which is interesting, although I don't know what to take away from that. It could be because the misfolded deadly version of the prion needs some starting material like the healthy prion in order to make enough misfolded prion to actually spread. The prion is first trafficked to the lymphatic system by cells in the intestine, and then from the lymph we may have neuroinvasion through nerves connecting to the gut. Once in the nerves, my next question would be, how do they spread from nerve to nerve? And this section is my favorite. It is, of course, the most related to what I do in terms of, thinking about cells and molecules instead of whole anatomical connections. But before I proceed, I'll answer Artistic's question. Does the immune system get activated by this prion-killing neurons? So neurons do not produce a very noticeable immune reaction. I don't actually know to what extent that matters, because here's the thing. The prion protein only causes disease, as far as we know, once it's reached the brain. And when it's in the brain, the blood-brain barrier keeps a lot of immune cells out. If the brain is damaged enough, the blood-brain barrier will leak, and that damage is probably going to recruit the immune system. So I'll say, I don't know for a fact, but from what I understand about neuroimmunology, unless the blood-brain barrier is severely damaged, those immune cells are probably not going to care too much until things are too late. I don't really know if there is neuroinvasion of the immune system in prion diseases that don't involve, compromising the blood-brain barrier. I will leave that open, although I don't know that for a fact. I'll put it that way. How does the blood-brain barrier keep immune cells outside? So I'm not a BBB expert, actually, but I think it's just a physical, actual, you just can't squeeze in. Although someone can correct me if I'm wrong on that. If we have healthy prion in our gut, how come we don't see a failing in our gut function if the prion is only needed in the wild type, I'm guessing. So my hypothesis there is that there is enough turnover of the gut where it doesn't really matter. We do see that prions do accumulate in the appendix, so it's possible that there are mechanisms by which we can just shuttle and quarantine the prion protein within our bodies and not have it be a big deal. But the problem with the nervous system is, of course, there is very, 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 very limited regeneration of neurons. So for the most part, once you lose neurons, they're gone forever. For the most part. The capillaries have particular structural qualities that result in the blood-brain barrier. Thank you so much, Marlon. Okay. So, how do prions spread from cell to cell? So here's a paper that I found. Prions hijack tunneling nanotubes for intercellular spread. This is really exciting because this connects to something that I learned for the first time in a different stream. So if you've been around for a while, you might remember that I talked about nanotubes in this stream. So this is the stream where I talked about the brain microglia, the macrophages of the brain. They actually slurp up these Alzheimer's proteins, right? So they use these little nanotube straws to connect microglia to neuron, and they suction the Alzheimer's protein right out of dying neurons. So again, thank you to all of you who make it worth coming back and make it worth me learning new things every day. I did not expect to see tunneling nanotubes appear in my reading list again so suddenly, but apparently, according to this paper that I can't show on stream because of copyright purposes, tunneling nanotubes might be responsible for neuron-to-neuron transmission of the prion protein. So that's really interesting. That's really, really cool. What else do we have here?
Yeah,
so I want this. So this is from that paper that I covered in that video. But if you didn't know, a tunneling nanotube is this super super tiny structure that we see here, that is like a couple of microns in diameter, that microglia use to suck out bad protein and to donate healthy mitochondria to neurons in need, and to other microglia that are dying. So these tiny little tubes that I learned, and I thought they were really cool, and I didn't think about them ever again after that stream. They are coming back again in a major way in the prion story, which is so incredible to see. I love that. Here's a hypothesis, I think unfounded by the time this paper was published, but this is the author's kind of spitballing. This is a process that I was completely unaware of. This is called trogocytosis. This is what happens when an immune cell nibbles on an antigen presenting cell. So in red here you have an immune cell, and in blues here you have an antigen presenting cell. And what we see here is actually the red cell nibbling at the membrane of this cell. So there is a hypothesis that transmission might be partially explained by cells nibbling on each other, because we know that the prion protein is a membrane protein. So nibbling at the membrane might be a way by which we can move misfolded prion from cell to cell, which is really, really interesting. The Zuck, exactly. The cellular Zuck. Nuts. Absolutely nuts. Very, very crazy. And this is sort of my hypothesis, which is, what about synaptic transmission? I didn't see anything about synaptic transmission in the review paper, which means this is probably not the answer, but I still want answers about this particular hypothesis, is that neurons communicate with other neurons through synaptic transmission, right? So one neuron releases neurotransmitter to the other neuron, and we know that the prion protein in neurons is localized at the synapse. We know actually that there are aspects of synaptic transmission that need the prion protein to function normally. That's not true for all regions of the brain, but we do know that in certain cases, if you screw with the prion protein, you screw with how neurons talk to each other. So by transmitting neurotransmitters, well, how does that happen? A bit of the membrane of the neuron blebs off and enters the synaptic space, and then that membrane merges with the following neuron. And since the prion protein is at the membrane, I'm wondering if it's possible that sneaking in with neurotransmitters like dopamine and glutamine is a little packet of misfolded prion. I don't know that this happens. I don't know if this is legit, but it's a hypothesis that I had, and I really want to know whether or not people have tested for that kind of thing, or people have observed that kind of cell-cell transmission. Small clear vesicles versus dense vesicles. I don't know if that is a way that you could look at it from like a gross anatomy perspective, but I would probably I would either try this in two ways. I would either engineer a mouse with a GFP tag at the end of the prion protein. I don't know if the prion protein will still fold correctly if you add a GFP at the end, but that's what I would try in order to track the prion protein in the cell, label it with something fluorescent. To do it with antibodies would require the cells to be dead, which is not really ideal if you want to do this in a system that is alive. So labeling genetically might be one way to test that. I thought neurons are only doing signaling through potassium-sodium ions. So that is an action potential. So that is how the signal starts from the one end of the neuron, and through potassium and sodium channels, you get propagation of that signal down the neuron. Synaptic transmission is what happens when this neuron tells the neighboring neuron to fire. Yeah, so it's two different types of signaling, right? One is just the action potential, which sends signal down the neuron. Synaptic transmission is what happens when this part of the neuron talks to that part of the neighboring neuron. Might be a bit of an oversimplification, but that's the way that I think of it. So that is actually it. I will summarize once more for those of you who are just joining us now. In terms of answering the question or hypothesizing mechanisms, right? Because a lot of this at the time of these review papers is still hypothesis. So the blood-brain barrier, probably unimportant. Misfolded prion requires normal prion in the gut to spread, potentially by requiring a seed to start replication. Prion is first trafficked to the lymphatic system by cells in the intestine. So they're absorbed in the intestine potentially. From the lymph, we may have neuroinvasion through nerves connecting to the gut. And then once in the neuron, there are a couple of hypothesized mechanisms by which that prion spreads from cell to cell, all without the apparent need of a robust blood-brain barrier. So instead of sneaking through the blood-brain barrier, we might be getting prions snuck into the brain through the neurons themselves. So, of course, the nerves of the central nervous system don't ignore the blood-brain barrier, right? They got that direct hookup, and so we might be seeing transmission of the prion protein directly through the nerves and not through other means of entry of the brain. All right, so that is what I have today. Bit of a short stream, but I will go ahead and I will take questions, and I will just hang by to chat as we end the semester. If you're still in school, I'd love to hear how your semesters are shaking up. This is, of course, again, my last ever semester of teaching. Maybe I'll have a job later where I return to university to teach, or maybe I go teach high school at some point. But in the meantime, I'd like to really try making YouTube my full-time thing. And again, shout out to members for making me believe that is possible. So it's very interesting. Is a prion actively moved to the brain, or does it just stay in the body so long it ends there by dumb unluck? That's an excellent question, and I don't think that question can be answered in the way that you asked it. So active transmission. So let me break that down a bit. So active kind of implies a sort of intentionality of things. So I will say that from the review paper, we have seen mechanisms by which you can get transmission from intestine to the brain. So I don't really think it's about buildup. It's just the prion protein ending up where it needs to end up for it to be moved, probably with a bunch of other things by accident. So it's not really luck, because it is going to happen at some point, right? But okay, so here's where I turn back the presentation. So let me go ahead and reload the presentation, or let me try to open up the thumbnail for this actually. Yeah, because I think this is kind of an important thing to acknowledge if I were to sort of summarize the anti-fear message that I have going on right now. So let me go ahead and enter my 1,000 folders where I keep all of my assets. And come on, OBS, don't crap out on me now. Not in the middle of stream. Yeah, because I want to. Okay, so here we have like this stock photo of general human anatomy. And let's put this below these two layers. Yeah, there I am. Welcome, Azure. Welcome. I joined late. Could you re-explain the seed concept? Yeah, give me a second. I'll first do this little soapbox that I have, and then I'll talk about the seed concept one more time. Can I just move me? Hello? There we go. So, what was I saying? So in terms of actively moving to the brain and tying that into my message of, having a reasonable amount of fear of the prion diseases, right? Because prion diseases are very scary. They always get upvoted on Reddit. Who doesn't love a prion video because it taps into something so terrifying? But I think what we're going to learn is that there are a lot of different avenues by which prions can get shunted out of the brain or delayed for long enough until it becomes an actual issue, right? So, we already learned that macrophages can destroy the prion protein and that dendritic cells cannot. So you can imagine that for some certain population of individuals who have, maybe more macrophages, maybe more active macrophages, that they are more capable of stopping the prion protein from reaching the brain. You can imagine that people, maybe with genetic variations in their dendritic cells, maybe they are a little more conservative with holding onto the prion protein and not releasing them into the lymphatic system, right? You can imagine now that we have this, like, I'm not saying this is the route, but a potential route from gut to brain. Now that we have that laid out in front of us, we can imagine all sorts of different ways that individual differences can provide a barrier to entry to the brain. So yeah, I think macrophages is probably like a big one. So maybe if your macrophages are more robust, maybe you can deal with it a bit better. Maybe if you have, I don't know, yeah, I mean, I don't even know what I don't know, right? Because there could be so many other different interactions between the dendritic cells and shunting to the brain that can have interventions that the body can already do. And I think for me, that fills a hole in my understanding of prion disease where, yeah, if a lot of people were exposed in 1989 to mad cow disease and not a lot of people have the actual disease and it's been 30 years, it's possible that actually it's kind of hard for the prion protein to make it to the brain. Right, that might be true. It might just be default hard for the prion protein to make it to the brain. Because as a kid, I thought, oh my gosh, if I eat contaminated beef, I'm going to die in 10, 15, 20 years, for sure. And what I think these papers, at least to me, what they're suggesting is that maybe it's not that bad. That maybe for some individuals, for some reason or another, maybe the gut is a little more leaky. Maybe for some reason they have more or less, connection between the gut and the brain. Unfortunately, like, and that methionine mutation is like the biggest caveat, right? Like homozygous methionine at 129 in the prion protein is present in basically the entire first wave of mad cow disease. So folding is going to be really important, right? Misfolding of that prion protein. And it's possible that if you don't have that methionine, or if you only have one copy of that methionine, maybe that's enough to really stop that prion protein right at the gut level and not really make it to the brain, right? You can imagine that if you have one bad copy of the prion, then maybe it slows down prion buildup enough for macrophages to take care of most of that protein. Maybe that's optimistic, but I would prefer to have an optimistic outlook than a dooming outlook, just for my own mental health, really. Yeah. Can the brain try to fight back? The brain's mostly focused on doing brain things, so I'm not saying that that's impossible, but I would be surprised. It probably is going to lean on the immune system to do more of that stuff. And I go back to answering Azira's question. Can I re-explain the seed concept? Of course I can. So I was saying how engineering approaches that require cutting the prion protein up into different chunks might not be the best situation, because cutting up the prion protein has been shown to provide more nucleation sites for prion misfolding. So you can imagine, I forget who said it, but it's kind of like the Hydra, right? You cut one head off the Hydra, it sprouts into two. So if you cut the prion protein, one strand or one misfolded protein into two, now you potentially have created two spots where the healthy prion protein can bind and misfold onto. So if you're going to take the approach of destroying the prion protein, you should do so very thoroughly so that you cannot get renucleation of the protein, which is why ion 717, which you can hear about in my prion video, again, go search through my video tab. That video tab, the video that I made will explain this drug that is currently in clinical trials. So ion 717 is an antisense oligonucleotide, very fancy word for a piece of genetic material. I don't actually remember. I think it's probably RNA. This is an RNA that binds to the prion, the healthy prion protein mRNA, and it prevents the healthy prion protein from being made so that it cannot be converted to misfolded prion protein. So that is one way that you can slow the progression of the disease, is just by stopping the healthy version of the protein from being made entirely. So I think I prefer that approach to an approach that engineers something like a chaperone or a protease, because unless you are grinding up the prion protein like into tiny bite-sized pieces, you might only be making things worse. 60 years is a pretty good life. Yeah, I would say so. If mad cow ends up being a disease that requires 60 years of incubation, I mean, is that sad in terms of like how long humans can live? Of course it's sad, but I mean, it's not as devastating as eating contaminated beef and dying 2 years later, right? Like that is the worst case scenario, essentially. Why do we not see prion diseases in sauropods, amphibians? Is it a mammal only? So it kind of depends on what you mean. So the mammalian prion protein is, of course, by definition, a mammalian problem, right? Birds have their own prion protein. I don't actually know to what extent the rest of the animal kingdom has prion proteins as a class of proteins, right? But in terms of the prion protein, only mammals have it. Other animals, like in the fish kingdom or whatever, they have their own version potentially, and I don't actually know if they get the general form of prion diseases. It's an interesting question, and it might just be because that their version of the protein is less prone to misfolding. The thing about the prion protein is that it has such a large surface area for misfolding, a lot of wiggly parts that can collapse into these tightly bound beta sheets that are really tough to break up. Like that is a unique feature of our version of the prion protein. I don't know if it is also in birds. It's possible, but in terms of answering the question literally, like why don't amphibians or birds get mad cow disease, for example? Well, they don't really make the protein that we have, is the simple answer. Do we see prion diseases in plants? So we see prion accumulation in plants, and I don't know if that translates into disease in plants. I'm of course not a plant expert, although Nep is Nepenthes VT, my fellow VTheorem member. It'd be kind of interesting to ask the question of if we can see disease in plants. However, my guess, Seon, is that we would probably not see prion diseases in plants, considering prion diseases need to be amplified in order to really be a problem for tissue. So if a prion enters a plant, well, the plant doesn't have a prion protein to, convert. So my guess is that they just kind of hang out and are pretty innocuous in the context of a plant. Again, I guess here. Here's a problem with regular neurons. They're too silent, something which does allow the brain to brain, but something like rabies infects the brain that is just over. I mean, that's why we have vaccines, right? Vaccines can stop rabies before it even gets to the brain, and that's pretty incredible, right? There are trade-offs, because if the immune system were very active in the central nervous system, you can imagine that would be very bad. Like, every time you got a cold, you just die because you get meningitis. Like that would be really bad. You don't want that to happen, and so you do want a robust separation of nervous tissue and immune tissue. It's not perfect, right? Because even through all the separation, you still get exceptions to the rules, like herpes viruses and rabies viruses love entering the brain anyway. But if any old disease got near the nerves and then you just lost function in your arm, that would suck, right? It's really, really important to keep the nervous system away from the immune system as much as possible. Does it affect all mammals or only eutherians? So I am not enough of a touch grass biologist to understand what a eutherian is. I'll say that prion diseases have been observed in quite a few mammals, and even more mammals have the prion protein gene, although I don't know if there have been recorded instances of prion disease in a broad class of mammals. I wouldn't know that. Didn't they just release a fancy cancer vaccine in the UK? I think they're still working on it, or they're ready soon. I don't think it's out yet. It's promising that mRNA vaccines can tackle cancer. It's very exciting for that kind of thing. Oh, it's marsupials and placentals. Oh, what does that exclude? I'm sorry, I'm very ignorant on animal biology, honestly. Because I kind of assumed that we have placentals and marsupials, and that was kind of it. Talking about proteases in your prion videos reminds me of the lichen proteases. Because of the multiple nucleation sites, is studying lichens for anti-prion treatment not worthwhile? So if I had to redo the prion video, I would maybe edit that part, given what I know today about nucleation, right? And like the fact that proteases are not strictly the only thing that you would need. However, if you were to combine like a lichen protease with other proteases that can work together synergistically to completely break down the prion protein, I think that might be better. Maybe I'm changing my mind. Maybe protease research for prion stuff is still pretty good, considering you can just, if you break the prion protein into 2 with one protease, and then you add a different protease that breaks that into 4, but 4 non-functional fragments of the prion protein, I think that would be pretty good, actually. I take it back. Platypuses and echidnas. Oh, interesting. Oh, very, very cool. Oh, I love having a chat that's so knowledgeable. I see. Yeah. I'll say that platypi and echidnas do have the prion protein, but they look very different than ours. I don't know if any of them have been observed to get prion diseases, especially because you can imagine that, like, why do we know that prion diseases happen in cows and sheep? Well, we raise them, and so we have human eyes on millions of cows and sheep every day, and so they could get prion diseases, but like, we're not raising platypi for like, we're not raising echidnas for food, essentially, right? We don't have that much of an investment in artificially breeding echidnas to the same degree that we would have for a cow. So I don't think that it is impossible for an echidna to have a prion disease. I just think that if it did, we would probably miss it, in all honesty. You'd also have to take into account that, like, prion diseases are not the only neurodegenerative disease. But also neurodegenerative diseases are also quite rare, just in general. You kind of have to live long enough to get one, and so longer living animals are going to have more time to have degeneration happen in their neurons. So I don't know how long platypi and echidnas have. I just kind of assume that, like, smaller animals don't live as long, which could be a huge overgeneralization. I just don't know. Or rather, smaller mammals, I think, just because of, like, R versus K selection or something. Talking out of my ass because I'm not an ecologist either, but like, that is my general impression that smaller mammals don't live as long, although please feel free to correct me if I'm wrong. I would like to say that I know absolutely nothing about 99.9% of what happens in the cell. I think it's a very worthwhile investment to get into, though. If you love Cells at Work, which I've heard a lot of people say that I should make Cells at Work content, and I feel very complicated about that because Cells at Work is so good. It's a fun fact that, like, once I started doing YouTube, I actually stopped watching Tirzu because Tirzu is so good. And I didn't want to be influenced by how good Tirzu is. So, like, revisiting Cells at Work, I feel like might affect the way that I do my YouTube channel, which I'm kind of afraid of. Maybe one day I'll do it. I'm not totally taking it off the table, but to all of those of you who have been asking if I've heard of Cells at Work, I have. In fact, this whole project came out of the idea of wanting to be Cells at Work, and then I got scooped at Cells at Work. So I actually am very painfully aware of Cells at Work. It is very, very good. Very, very good. What biological problems would you want to research if given $1 billion? $1 billion, it's a toss-up. I mean, this is such a low-hanging fruit answer, but like cancer, like obviously it's got to be cancer because, well, it's cancer or Alzheimer's. I don't know which I find to be more tragic, right? $1 billion, it's tough. Okay, so I'll say 3 answers. Please don't ask me to pick one because I love all 3 of these problems. It's Alzheimer's because of its prevalence and because of the tragic human cost, right? It's, any human dying is of course a tragedy, but in some cases the mind of a human going before the body is just another layer of tragedy that I find to be really horrific, right? If you've ever had a loved one that has had dementia, it can you know how like heart-wrenching it is to not only for the lived family of the patient, but also for the actual individual going through Alzheimer's. They are scared and lonely and depressed and angry, all the negative feelings while they are essentially losing their identity. And that is, of course, not a fun way to go. So that would be one of my answers if I had $1 billion. My second answer would be, of course, cancer, right? Cancer is extremely common. You will either have cancer or you will know someone who will have cancer. And, you know, anyone who's been to my glioblastoma stream knows that, like, how personally tragic I feel cancer to be. I like cried on stream. I like lost it every time I like go back to that paper in pediatric cancer, just because, you know, you can live your life completely normally and do all the right health things, and you still get cancer, because cancer is a combination of genetics and chance, essentially, and factors that we don't fully quite understand, and can take people before they are before they have gotten the chance to really live. And I think that's really tragic in a different way, right? Because Alzheimer's for most people is quite late onset, although there are cases where there are early onset Alzheimer's. Again, another layer of tragedy, but like cancer is yeah, it's bad. You know, I don't think I convince anybody that cancer is bad, essentially. My third answer, if I had $1 billion, would actually be climate change. And you might be wondering, why is that a biological problem? Well, it's because biology has great untapped potential for producing cost-efficient solutions to climate change, right? And I think that I will eventually go down into that rabbit hole, but I think I'm having too much fun talking about pathogens for now. But like, you can build huge reactors that are reliant on things like electricity and power and machinery and stuff like that. But at the end of the day, nature has had many millions of more years to innovate on potential solutions than humans have had. And of course, to harness the power of microbes is, I think, really, really going to be a big way that biologists save the whole universe. Because what is cheaper, building something that sucks, you know, CO2 out of the air, or being the one person that makes plants a bit better at sucking CO2 out of the air, right? Like, well, if you can engineer a plant that's just better at doing that, then what's the cost of planting more plants? Like nothing, right? Comparatively to building a huge machine. And so yeah, it's a toss up between those three, because science is forward looking, and, you know, we need to care about more than just our immediate generation, right? And so actually, if it were me, that might be the answer, because there's already so much money going into cancer research and Alzheimer's research that maybe a place that I find to be deserves more attention is the utility of biology in making sure that humans get to inhabit the planet for a lot longer. Yeah. Dude, I'm an engineer. I only know enough biology to do my job. That's kind of implying that you are using biology in your job, which I think is really interesting. Do you want to share a little bit more about what you do? They made me feel pity for a cancer cell. Yeah. You know, if you watch my cancer stream, my glioblastoma 12-hour stream, you can kind of think of cancer as cells that like escape the matrix, right? Like, your duty as a healthy cell is to self-eliminate if you are going to pose a danger to the germline. And in some ways, like, if you want to ascribe poetry and narrative to the body, you kind of can't blame cancer for just wanting to exist. But from a narrative perspective, it also makes it very clear that when you have like a cell that is cornered like that, they're going to do everything and anything and everything to get out of that corner. Yeah. Fun fact, I was diagnosed with brain cancer at 9. I'm 30 in November. Congratulations for beating it. That's amazing. That's awesome, especially because the odds of that are very low. And so that's incredible that you've been able to uh, grab a lot more of life than what was statistically probable. It's really amazing. What do I think of the Ion 717 trial for prion diseases? So I covered that actually in my prion video, if you haven't checked that out. And I think it's a pretty good idea, right, to stop the production of prion at the level of mRNA. But I think that we could do better, because that is slowing down the problem, not necessarily stopping the problem. So I think we can do better. Do you have any personal hunches on biochem problems that you can't exactly prove yet? Oh, so do I have any hypotheses that I do not know the answer to? It's an excellent question, Mies. I'd have to really think about that, I think. Yeah, I have to really think about that. I think that I'll say this very generally. So nothing really comes to mind right now, but I'll say this. I think we are going to be a lot more cognizant of how the immune system and the nervous system co-function, and that is going to explain a lot of neurodegenerative diseases. I think that the amyloid beta hypothesis of Alzheimer's disease has had its day, and I think that microbial influence and viruses, I think, are going to have more impact on understanding the full picture of how neurodegenerative diseases happen than what is currently appreciated. I don't want anybody to freak out about long COVID or anything. I just think that, like, through studying viral infection of the brain, we might get a better understanding of how the immune system functions. And if we get a better understanding of how the immune system functions, that is going to enable us to think about how neurons are not being serviced correctly in those diseases. This is, of course, my bias. My formal training was in neuroimmunology, meaning my whole thing is that I think about brains and the immune system. So I want to be very vague there, that infection and damage mechanisms, I think, are going to be a lot harder in the future. Yeah, at some point I'll talk about Zika too, because I did quite a bit of reading on Zika when I was in graduate school. If plants could be more efficient, wouldn't it have evolved already at some point? So plants, okay, so this is more like an evolutionary philosophy kind of thing, where, you know, things shaked out the way they shaked out due to chance, probability, and mass action, right? So the way that plants got around making a pretty crappy protein, although there's a recent paper, I think, that came out that argues that Rubisco isn't that bad, which I think is something that I will read for the future. So Rubisco is a carbon-fixing enzyme, carbon-fixing protein in the plant, and researchers are losing hair trying to improve Rubisco, because if we could just improve Rubisco, that would make carbon fixation a lot easier for plants. The way that plants evolved to get around making such a bad, quote-unquote bad, again, there are opinions out there that Rubisco isn't that bad, but the way that they get around it is to make a crap ton of it, to just make a ton of the Rubisco protein, so they can kind of get around not needing to evolve a better protein because they can just make more of it. So it's impossible to say whether or not there's an alternate version of the universe where the first successful organism that made Rubisco did it in a more efficient way. Maybe that would have caught on. Although mass action suggests that maybe there's some fundamental engineering aspect that is preventing Rubisco from being better. But I would not count us out yet, because molecular biology is only as old as there are people alive today that are as old as the field of molecular biology. And while it has been frustrating and hard for scientists to engineer a better Rubisco, this field is so young. Like, if you compare it to physics has been around forever, right? And like, I think it's better to compare molecular biology to quantum physics, right, which is relatively recent in the timeline of things. And I think that it's not a reason to lose hope, right? I think because we are so young in this field that we've been able to do, like if you think about it, right? Like, when was DNA visualized? It was like, I don't know, like 70 years ago, something like that. We went from not knowing what DNA did to trying to engineer a protein that nature has failed to engineer for millions and millions and millions of years, all within the range of 70 years. We got big old brains. I think one day we'll do it. I spent the money to 3 parts, 500 into period cramps, and so much pain and very few attempts to reduce it. The rest goes into TB and Alzheimer's. Very, very, very good choices, I think. I do think that, like, especially now in the United States of America, like, women's health gets not as much attention as it deserves. And so if you tune into my next 12-hour stream of Ganza, I think I might do a whole day where I look at I'll call it female specific biological problems, because I think it's not to say that, like, females are more special than males or anything like that, but it's rather that there are things that are associated with being female that are different than males, and traditionally medicine has been focused on the male experience. And so if period cramps are something you're interested in, you might also be interested in other aspects of female biology that I think are worth talking about and are interesting. Does the prion protein have use in the body? Studies suggest that it has something to do with myelin sheath maintenance and synaptic transmission in certain scenarios. So it has some sort of function in the nervous system, but it's not entirely clear, put it that way. I'm a environmental engineer, and besides typical stuff, we also study ecology, microbiology, and toxicology. Yeah, so Cerium, that's going into what I was saying about harvesting the properties of microbes for climate change. So I think we would have overlap in that regard. With Microsoft's quantum computing chip, do you think protein structures will be completely discovered? You always gotta confirm things in vivo. Like, I know that there's all sorts of hype with AlphaFold and computationally solving structures, but you still gotta test it. Like, there's a huge push in the US, which is super annoying, to outsource a lot of wet lab stuff to AI, which is frustrating. But at the end of the day, like, so here's the thing that will never be replaced by AI in terms of so AI is only as good as the cumulative current understanding of the world that humans have, right? So AI cannot make a perfect prediction if it does not have all of the data. And until we know everything that happens in the cell, AI will not be able to give us 100% confidence in what proteins look like in the cell. Sure, you could fold the protein in a computer, and maybe that'll be the same way that it happens in a cell, but it also might not be, right? There are processes in the cell that can alter the structure of proteins. The protein might be in a pH specific environment that could change things. You can have post-translational modifications that can change things. You can have localization to certain membrane compartments that can change things. You can have modifications that we don't even know about changing things. You can have interactions with other proteins that may be artificially folded in some different way, right? We have like chaperone proteins that are important for folding, and maybe interaction with certain proteins cause certain proteins to fold in ways that would not be computationally predictable based on their amino acid sequence, right? So I'll say that I think AI can do a lot, like a surprisingly amount of good, robust predictions for protein structure. There's like an impenetrable barrier, in my opinion, that AI will never breach because it requires us to know more. And unless we have a perfect knowledge, the AI will also not have perfect knowledge. My opinion, of course. Other people might disagree. Yeah. Evolution doesn't even select for perfect, it selects for good enough too. Exactly. So evolution, it worked enough for plants for it to happen. But it's also possible that maybe a better Rubisco would be worse for the plant in some reasons, and maybe we need a little more engineering on the plant side for the plant to tolerate a better Rubisco. That's also possible. And that's something that Miesman is saying. I feel like engineering a better Rubisco would be really dangerous. If it escaped the lab, it could be one of the worst invasive species ever seen, depending on how much better it was. There would have to be a lot of fitness testing for this particular thing, right? Like, sure, there's always a risk for something leaking into the environment, but tides are rising. Tides are rising. Places are drying out. it could lead to a disastrous loss of biodiversity. It might also not, right? Like, as I said, there might be a reason why nature chose to make like a lot of Rubisco instead of making Rubisco better. There might be some unknown penalty that the plant might have if Rubisco functioned better. We'll see. Also, you might not need the plant to grow Rubisco, right? Maybe you just need the Rubisco protein. I don't actually know enough about this science to give you a confirmation. If you could, for example, if you could build an artificial plant, right? Something that doesn't make seeds, for example, right? A sterile plant that exists to make Rubisco and to just eat, to eat the sunlight and make oxygen and recycle that carbon dioxide, but not actually make a seed, right? That would be really interesting, wouldn't it? Yeah. Sorry, going down the chat now because I'm behind. DNA was first seen in 1990. I think it was 1890 under the microscope. I'd be really surprised if it was 1990. I don't think that's true. Yeah. Is the prion protein held in common between BSE, CVJ, and Kuru? Yes, that is the same prion protein. BSE is the variant that comes from cows. CDJ, I don't know what CVJ is, but CJD, sorry, CJD. CJD is the one that comes from humans, and Kuru is also from humans. I don't know what CVJ is, though. Only just got here. Hey, Clockwork, welcome. Hope it's a good stream. Wish I could have read the paper on time. I mean, I rushed both papers anyway. Essentially, I'll summarize it real quick for you. Prions enter the gut through the intestinal cells, either M cells or enterocytes or dendritic cells, get shunted via nerves to the CNS, and then get snuck into the brain via neuron-neuron transmission, probably, right? All of that is hypothetical. But what's interesting is that the blood-brain barrier is probably not involved, which I think is really interesting. I feel very skeptical about AlphaFold. I think there's a correct amount of skeptical to be where you can be very excited that you can have a lot of robust structures. It's just you just have to do the thing and test it, right? I think that's fine. Never be satisfied with just an AlphaFold structure. Take the AlphaFold structure, make a prediction, test the prediction. That's what you need for AlphaFold to be meaningful. Got a friend dismissed from ER once because of period cramps. Turned out they had endometriosis. This is why I love teaching so much, and I'm actually very sad to be leaving my job, or rather kicked out of my job. We actually had one of the final projects in my class be about trying to have an early detection method for endometriosis. I'm not going to say what their idea was because I think the idea is quite nice. There were issues with it, sure, but but I liked their idea, and it is a novel idea. No one's ever tried it, so they deserve to have that secret. But what's so lovely about my job as a biology instructor at a university is that I get to see the percolating minds of the upcoming generation. And this class has been the best class that I've taught so far. And I don't feel shy about saying that because who's going to rat on me, right? Who's going to be like, oh, they liked this class more than liked our class. I'm going to complain while the university is firing me. So like, so they can complain to nobody, right? This particular class that I'm leaving right now has brainstormed and created a project. They didn't test it, but they came up with the idea of an early detection method of endometriosis because it's very difficult to diagnose. And so I think it's nice, CDM, to be aware that there are young scientists, budding scientists out there, only 19, 20 years old, who have come up with pretty interesting strategies to tackle these issues that have been neglected. I haven't seen studies verifying its prediction, plus intrinsically disordered proteins. I mean, intrinsically disordered proteins are not going to even have a crystal structure. They are disordered, so those floppy regions are going to flop, right? And I don't think that's a big deal. I haven't seen studies verifying its predictions. I haven't either, but I haven't really looked. I'm sure they're out there, right? Because you can make not structure predictions, but rather prediction. So say, for example, you have a protein that is hard to crystallize and you have a drug and you fold it with AlphaFold and there looks like there's a pocket that, a certain drug binds to, right? You do the AlphaFold and you get a binding pocket. And then if you use that drug in like a cell system, and you find that in an artificial system that it cures the disease, that is an example of a way that you could validate the AlphaFold system. Here's what you don't want to do. You don't want to fold the protein on AlphaFold, design medication, and then use it on a human right away. That's what I mean. You don't want to do that because you have to first test it. AlphaFold can help you design experiments. It does not replace experiments. Yeah. I feel like AlphaFold is just a machine built to do something with not enough instructions to do its job. I'm really surprised I have so many AlphaFold skeptics in my chat. AlphaFold is really, really good. It won a Nobel Prize. It's just not perfect, but it is really, really good. And this is something that I want to make sure that my audience is aware of, right? Like, there's a level of skepticism that I think that is very easy to have, where if something in science is not perfect, then it deserves a ton of scrutiny. And I'll say it again, AlphaFold is really good. It's just not perfect. And I don't want... I want you to keep that skepticism because the spirit of skepticism is very important. But I don't want to head down a path where I am cultivating an audience that is hyper skeptic, right? AlphaFold still won a Nobel Prize. It is very good. It's just not perfect. That's all. Yeah. You need carbonic anhydrase to convert the dissolved CO2 into something Rubisco can actually use. Carboxysome is a giant bag of carbonic anhydrase around Rubisco. So yeah, maybe a completely in vitro system isn't great, but maybe sterile plants could be a possible engineering strategy. I think that might be worth thinking about. Hey, Cleonium, welcome. Yeah, I'm so behind in chat, sorry. I feel like I've heard people say things like we share 50% of our DNA with bananas and like another arbitrary number throw out to say that we're close to monkeys, but I feel like there's some context missing. I mean, a lot of our genes that we have are essential to the very act of being alive, so it's not surprising that we share a lot of our sequences. However, the parts that are refined to make us human are relatively low in number, so that's kind of the disconnect, I think. Very proud of the kids, but I also feel like I'm pathetic. So I can't give away what university I teach at because it would dox me. But like I when I think about the place that I've taught at, I am filled with so much pride. And I know like Doobie like revealed what she had done in her IRL life, and how excited she was to say that she had been an Emmy nominated the Foley artist, right? So if you don't know, Doobie is a very prominent indie virtual YouTuber, and in a past life, in a past VTuber identity, she had not been able to talk about her real job, and when she became Doobie, she had revealed that she was a Foley artist, an Oscar-winning Foley artist. Like, so for me, like the location that I'm teaching right now, to me that is the equivalent of like winning an Oscar for Foley art. But it kills me that I cannot share right now what university that is it. That is not to say that there aren't fabulous, brilliant students in other universities, right? But like I am just expressing from my loyalty and honestly a bit of my shared identity with this university how proud I am of this particular class. It is really, really incredible what I think they will do in their careers. If the US won't have them, I would be very hopeful that there will be a country that does take them, because they have great ideas for being so young. And not like Elizabeth Holmes, right? Not pie in the sky. I checked their ideas. Maybe it won't work, but it won't work because of some very niche biology, some very like specialized biology, not something ridiculous as a homeopathic amount of marker in a drop of blood, right? Like they have ideas that work on paper, and that's incredible for being 19. Yeah. Yeah, unfortunately, I'm sorry, Scarlett, you did miss the stream. We're just kind of chatting right now. Endometriosis is a tissue structuring disorder in which tissue resembling the endometrium forms in parts of the body that it shouldn't. Exactly. Like you get what's called ectopic, meaning inappropriate location. That's what ectopic means. Development of tissue that should not be there. Is this going to be uploaded as a video? This is going to have a VOD, so it's just going to have, it's going to be in my live tab as a recording of the stream. Yeah. I know what you mean. I tend to be very biased pretty heavily against anything AI. Yeah. I mean, there's good reason to be skeptical, but in terms of computational modeling, it's pretty dang good. Hi, love your vids. Thank you so much, Scarlett. Thank you. Do I teach at a public university? I'm guessing that's what PUI means, or an R1. I don't want to like give away at all where I teach. Unfortunately, I like maybe in a couple of years where like all traces of me working at this university are gone and then I can't be doxxed anymore. Maybe I'll share that. Maybe I'll share then. But like, yeah, to me, it's like the equivalent of winning the Oscar. I also applied to this school a couple of times and got rejected every time. So it's like, it's always been a dream of mine to be at this university. Even though they didn't want me as a student, I'm very, very honored that they had me as a teacher. Yeah, and it's every bit as wonderful as I expected it to be, put it that way. Primarily undergrad institution. Yeah, I'm still not gonna reveal that. Still not going to reveal that. I don't want to narrow down the possibilities at all, except for I'm Omega proud and brimming with, just very happy with the quality of the students. For certain, all of the students that I teach are smarter than I was when I was a student, basically. Right? Like, sometimes it feels weird to be teaching a student who is actually smarter than you, right? I've actually had that, so smarter, not smarter, that's like an oversimplification, but I think at this university I encountered maybe 2 students who have the amount of biology knowledge and skill that I had. And that's insane because again, these are like 19-year-olds, right? 20-year-olds. And the fact that they have consumed enough content and refined their thinking to the point where I think that they are, the only thing that I have is years of experience on them. But like for some of them, they have reached my level essentially of like, so I'll put it this way. If you were to put me in a room and them in a room and had us both come up with a research project, there are 2 students that I think would do as good of a job as I would, which I think is insane. So I might have more years in terms of just, I know more things. There are 2 students who already have more skill than I do in terms of biology, and I'm sure that like everybody that graduates this university is going to be better than me very, 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 very quickly. But it's the first time where I was like, they walked into class and I was like, oh, you are already better than me, and that's insane. I don't know what I have to teach you other than how to write properly, because some people it's difficult to write. Yeah. What if your student's research becomes famous and the headline reveals the university too? I'm sure they will. I'm sure these students are going to be some of the best researchers in the world at some point. But you'll never be able to trace it back to me, I think, unless I choose to reveal like, hey, this was my student. Then you might be able to find me. But I value my anonymity. Related to what you were talking about with climate change, I feel like biologically degrading plastics is the only real viable solution to the plastic pollution problem. I certainly agree. Yeah, mechanical and chemical, that is the power of biology, right? Is that you can have large scale efficient solutions that are programmable in a certain way. And I think really, I mean, I don't know, right? Like, I'm not a physicist or a chemist, right? Like, I don't know what it's like in those fields, but I can say that like it's really, really cheap to just grow bacteria in a vial. Now, is it expensive to do like in terms of like relative cost, right? In terms of the amount of dollars that you need to roll out a bacteria thing, that like maybe you have a bioreactor, right? Like maybe if you're worried about environmental contamination, you have landfills that are physically blocked off from the environment, and you dump in some plastic degrading bacteria to form a bioreactor, and you feed them the plastic and they output carbon or something. That would be awesome, right? Like, and you would do that, and the bacteria are self-feeding, so they reproduce, but they're contained in this physical environment. And honestly, if a plastic eating bacteria gets away, it's probably so focused on making plastic degrading enzymes that's probably not going to outcompete the tough and hardy soil bacteria that have existed there for centuries, right? Like, I think that environmental escape is more important if you don't have a dead man switch, which, by the way, in certain not metabolic, but microbial engineering projects, you actually do have genetically encoded dead man switches, where if you drop below a certain threshold of the thing that you are trying to ingest, say plastic, then that triggers a genetic switch that kills the bacteria. So when the bacteria are done eating the plastic, they all just blow up. Is it possible that you have escape from that? Yes, but statistically speaking, that should get rid of most of everything. Very, very, very cool genetic approaches in microbes. And eventually I will cover that in a stream. Like, the field of engineering with biology is really fantastic. Hey, Bone. I don't know how much stream is left, but I'm excited to catch what I can. We're just chatting right now. We already talked about the prion portion. But essentially, for a recap, a lot of people in the UK have been exposed to prion diseases through mad cow, right? The scare in 1989. But despite so many people potentially being exposed to the prion protein, only like 180 people in the UK to this date have had a prion mad cow disease, which indicates that there's some barrier that is slowing down the transmission of prion protein to the brain. And the stream is all about a potential route which goes from the gut to the lymphatic system to the central nervous system, and then passed along neurons through things like nanotubes or cell nibbling, which is something that I learned today. And now we're just chatting about biology in general, which I am so happy to do, because I feel like when I don't stream every week, I get like antsy. Like, I love talking about biology. I love doing my videos, of course, but like it like editing on your own lacks the social aspect of what makes science so fun. And now that I'm not going to be employed by my university anymore, I feel like streaming is such a nice outlet for, feeling the hype behind biology, because students, as great as they are, have a grade, and sometimes they're not happy with the grade that I give them, so it kind of taints the experience of learning. But for you in my chat, it's I'm very grateful that you all show up to hear me yap about biology. Yeah. No interest in tracing or doxing you, but I really hope their ideas work out. Early detection for endometriosis would be a game changer. Yeah, it would be really, really nice for people who can experience endometriosis, because, as someone who cannot, it's just one thing that I don't have to think about, and for people who do have this, it's really painful. And pain reduces the quality of life by so much, and to catch it before it becomes painful, I think would be huge. And yeah, I really think that I'm very happy to have helped them refine their ideas, to challenge their ideas, right? Because it's not like they came up with their ideas in a vacuum and then came out with a really good idea. I got to be a part of the process that refined their ideas. I said no to a lot of what they wanted to do. And through this conversation, right, which is what biology is at a very high level. It's a conversation between you and more experienced researchers. You have, you as a young researcher, you have the moxie, right? The attitude, the hunger, the will, the sharpness, the quickness. And then you learn from, people that have more experience than your perspective widens, the possibilities open up, and then you take your youngness, your energy, your excitement, and then convert that into something that is actually very, very exciting, right? And to be the part, to be the wall that pressures them, right? Like I call the final project pressure testing, right? The first 2/3 of the class are me teaching you, and now the last third of the class is you teaching me. But in order for you to teach me, I need to pressure your ideas. I need to make sure that they withstand scrutiny, and that process is so much fun, right? That's why, like, I started this thing in my Discord, exclamation point Discord if you haven't joined, where you can submit one of your ideas, and I will do the same thing. Like, I will, if you're okay with your idea not being secret, of course, I will critique your idea, and you can experience what it feels like to have your ideas pressured, because I think it's very, very fun, right? It's like an intellectual game where, like, it's a cooperative game, right? We're both trying to make a good project, but you are throwing a ball to me and I'm throwing the ball back to you with revisions. And through that process, you can watch your ideas improve over time, and you can see the blind spots that you had missed before. And I think that's my favorite part of teaching is you have a young, hungry scientist and they have a great idea that's flawed. And then you help them iron out the flaws. And then you both have something that you can be very proud of. And I think that's very, very fun. IMO biology is engineering with chemistry. I think that's fair. Hey, Adri, welcome. Yeah, you run into some insanely smart kids. Some of the undergrads I TA for have papers while doing 12 credit hours for this semester. Chemists, by the way. Yeah. Some of these young people are insane. Much better than me. At the very least. I am so behind in chat. This is what Psy feels like, I think. I spaced out again. I would not pass your class. I've had students pass out in my class all the time. Don't worry about it. And they've passed. Because lecture is not where you learn. The place where you learn is actually when you do homework and when you do your assignments. Lecture makes doing your assignments easier, but the actual learning happens when you apply your ideas. Yeah. Is the difference between CJD and BSE simply their origin in the ecosystem? I think it would technically be the fact that bovines make a slightly different version of the prion protein than ours. So I think the answer to your question, Marlon, is yes, asterisk, with the focus on it being the actual version of the protein that we make, because we don't have 100% overlap between our prion protein and the bovine prion protein. Not only that, but there's a risk of ectopic pregnancy. Yes, exactly. Like if you have female anatomy, there are a lot of things that can go wrong in the female anatomy that I think require attention. And in this coming January, I am so excited to talk about that and to shed a light on some stuff that the fellas might not really think about. Yeah. Yeah. Yep, so there's the Discord. Yep. While I'm still live. Yeah, we're just chatting now. It's been a while since I got to chat with you all, and I love chatting, so. It's not just that the ideas withstand scrutiny, but also to be able to think on the spot and have confidence in said ideas, to be able to not fight back, but make valid points and promote creativity. Yeah, creative thinking. Yeah, and that conversation is really important, right? Like, they'll throw their idea to me. I will say, I don't know that this works because of what I know about biology. And so now I've taught you that missing piece. Now you have to go find a better idea. And I think that back and forth is so fun. Man, this biology thing must be harder than I thought. That's what every student says in this last third of the class, that like they all come up with their first ideas. They're all very excited about it. And I just have to be like, no, you can't just ask a bacteria to make any membrane protein. That was actually one of the suggested techniques for, I'm not going to give too much of the problem away, but like somebody wanted to engineer a bacteria to make a certain membrane protein that could have functionality. And I was like, well, here's what I know about making membrane proteins. Eukaryotic cells have Golgi and endoplasmic reticula that have particular pathways where you can make a protein that gets folded in the ER and pass through the Golgi to be trafficked to the membrane. A bacteria does not have that. Bacteria have what are known as secretion systems. So they can't really, as far as I know, bacteria cannot be programmed to make complicated membrane proteins. There is something called bacteria protein display, where you can engineer a bacteria to make very simple proteins at their surfaces, but nothing that is that complex. So that's an example of an interesting idea coming from someone with less experience, having to be refined. So whatever goal they had with getting this to work could not be done with making the bacteria make a complicated membrane protein. That kind of nuance I think is really interesting, right? Because this pulls from your collective knowledge of biology. It feels very last exam kind of thing, or practical exam, without actually being an exam. Because yeah, in the real world, you're not going to be sat down with an exam in your PhD unless it's your classes. In the real research world, no one's giving you tests. The real test of your knowledge is, can you figure out how to solve a problem, either a research question or an engineering product or something. And I think that is such a more valid way to assess a student's competency than a written exam. Although I'm not in favor of removing written exams as a whole. It's just, I think these kind of practical, like, solve a problem. How would you do it? That's a much better way to evaluate a student's ability than your typical test, I think. Yeah. I am so glad you've had a good experience with teaching. I have felt that I would enjoy doing that since I graduated. I got very, very lucky. And also Mylan's here. Welcome, Mylan. Mylan being my fellow V-Theory member. You can find them, exclamation point V-Theorem, a psychologist VTuber, and also the person that I go to for all of my statistics questions, because in your vanilla biology education, statistics is not very well taught. So I'm very endlessly grateful for Mylan's statistical talent as well as their artistic talent. So go check them out. Yeah, we're just chatting at this point, reflecting, I think, on the semester now that it's freshly ended here. I want to become a chemist. Do you have any good advice? Not for me, but I'm sure we have chemists in the chat. Would yeast be able to do that task as a eukaryote? So yeast have yeast display, and I believe, someone can correct me if I'm wrong, but I think yeast are better at it than bacteria are. I think bacteria are not great. Phage can do peptides. I think yeast can do it better. But I think, yeah, overall, asking a microbe to make a complicated membrane protein is not that easy. Although, if I had to pick, I think yeast are almost for certain better than bacteria at doing the same job here. Yeah, completely agree. Problem solving is so much better than route learning. And it's such a shame because I don't think it's appropriate to do that for every class, right? For example, like math classes, like how are you going to have a math practical, right? It's just, you just kind of have to have an exam. But I think a lot of biology can be moved into this like practical realm, right? Unfortunately, it would probably like, so if I had to restructure biology education for university specifically, right? Because I think high school, you gotta take the test. You just gotta memorize things. You don't get good at biology if you skip the learning. Like I was watching a martial arts video earlier today where this kung fu guy showed off how strong his hands were. Like they were like buff as all heck because he had been punching sandbags every day for years. And yeah, if you punch sandbags every day for years, like I don't care if you think kung fu is fake, right? Like the guy that punches a sandbag every day for years, if a punch from that man lands on you, it's gonna feel like a brick wall because of conditioning, right? And so every training montage and every shonen character, right? I just want to learn the techniques. I don't want to learn the basics. It's like, okay, no. Biology is the same way. Like you got to do all the dumb memorization, the hard memorization. You have to know everything about the ER and the Golgi because you have to have a familiarity with them. You don't have to have it memorized, but you need to know basically how the Golgi functions. You need to know basically how the endoplasmic reticulum functions. And when you need to remember what they do, you have to be able to remind yourself very quickly. You can't, it's an inefficient use of your time to have to teach yourself basic biology every single time you need it. For Journal Club, like I will relearn all the time because it's impossible to have a working knowledge of every process in the cell, but you gotta just do the memorization. It is the equivalent of punching sandbags when you are young. And then like how I would do it in undergrad is I would merge biology 101 with a lab class, which is, not too far than what we do for my class right now, because there is a laboratory component, but I would do it for the very first biology class, which is maybe not very practical for a university setting, because biology 101, like assuming you took biology in high school, biology 101 freshman year is pretty much the same, like very similar. There's more, of course, but like content wise, it's not that much more. And like, if you learned some lab stuff alongside with the fundamentals of biology, then by the end of class, you should be able to come up with your own research project. Or, you know, as much of biology should be geared towards trying to problem solve as physically possible without neglecting the sandbag portion of it, I think. Yeah. Did you have any students using ChatGPT to do their homeworks or exams? I graduated right before that became a huge issue. So here's the thing about ChatGPT for a lab class is that ChatGPT has to be able to learn and interpret data, which is hard to do, right? Because, you know, ChatGPT can take a text prompt, could probably take an image prompt, but data is data, right? Like data is just numbers. And ChatGPT, you know, if you had a prompt to ask ChatGPT to do your lab report, you pretty much did your lab report. Whether or not students have used ChatGPT to like write their introductions, I don't actually know. However, what I will say is that I have seen realistic growth from the beginning of the semester to the end. So at the beginning of the semester, when they were first writing their papers, oh man, every beginning of the semester is a disaster. It takes so long to touch up their writing because scientific writing is very, very difficult. And so, while I can't say that they didn't use ChatGPT or they did use ChatGPT, I will say they were pretty bad in the same way that all students are bad when they first start writing science. And by the end, they were writing much, much more clean. And I think I might have a stream one day where I teach some basics about scientific writing for students who are interested. But I feel like that's such a boring stream idea to convince people to show up, right? Like, I don't know, I'm trying to think of a thumbnail concept that would make scientific writing 101 like something interesting that people would care about. But I genuinely think that it is very, very interesting. I minored in English rhetoric, right? I minored in the art of language for functional purposes. So the difference between linguistics and rhetoric is that rhetoric is used as an end of something. So it's not the study of how language is constructed, but how to construct language for a purpose. And so I love like reading student writing, and again, like the back and forth of like helping them patch up their writing so that they become not only clearer writers, right? Because there's a feedback mechanism where if you write more clearly, you will be able to think more clearly, because by the end, you get a sense that the students' minds have been refined in a way where they are thinking in a more clear way. Not only are they communicating ideas better, but they can better come up with those same ideas. So I think that, to me, that's a great stream, but I find it hard to believe that people would show up. Although, let me know in chat if that sounds interesting to you, that if you would like to know the secrets to communicating in a scientific manner, or just how to organize your thought. Maybe that's how I structure it. Maybe I don't structure it like science writing 101, but maybe I structure it like how to organize your thoughts in an efficient or a scientific way, like a scientist. Maybe that's more catchy. Who thinks Kung Fu is fake? I mean, in terms of proportion of MMA fighters who have Kung Fu as their background, relatively low. So on the competitive stage, Kung Fu doesn't really have that many huge accolades. Not to say that nobody in MMA has like Kung Fu background. It's just not very common. It's a little more niche, I would say. Yeah. Once we had to build tiny ecosystems in terrariums at ecology class. Oh, that's so cute. I love Life in Jars, by the way. Another great YouTube channel where he just collects like pond water and lets them grow in an anaerobic environment for years. It's really great. As someone who doesn't know anything about animals, it's really fascinating to see the coming and going of certain species. You need to be able to wayfind through humanity's comprehension of biology when dropped in an arbitrary spot. Exactly. That's a great way to put it. Is that like, you don't need encyclopedic knowledge, but you do have to figure out your directions. Spurious Correlations has an AI that tries to explain unrelated data as if one caused the other. That's very funny. I'd show up. I'd want to see how you think about it versus how I was trained. Mylan, maybe we should just collab on it, right? Like, I think so much of my personal entertainment is seeing how different people are trained, right? Like having conversations with biologists who aren't molecular is insane. Having conversations with psychologists has been insane. It's part of why I love being in V-Theorem so much, is that our basic fundamental training sometimes can vary in very interesting ways, where we think we're all doing the scientific method, and we are. We're all doing the scientific method here. That's why we get to call ourselves sciences, right? Anybody who says psychology isn't a science can fight me and the rest of V-Theorem. But just because we follow the scientific method the same way doesn't necessarily mean that we were all trained with the exact same best principles. So I think it'd be really interesting to collab if you are down for that, Mylan. Although I think you're quite busy these next couple months. I'd be there for that stream. That's interesting to hear. To make a stream for science writing, it might help if you choose somewhat ridiculous topic to write about to make it more entertaining. Maybe. I'd consider it. Unfortunately, research field is very dry where I live because there is far too much of professors and mentors placing themselves as first authors instead of co-authors or correspondence authors. That's insane. I would what? I've never heard of a professor putting themselves as first author. So I'd be very curious as to where you live. Of course, although you never have to. You never have to explain if you don't want to, but I've never seen that be a adapted principle. That's crazy. That could be fun. Yeah, I think it would be fun. It would be fun, I think, to take something that is relatively neutral about because we've talked about doing VTuber research before. So Mylan, I think if we had a project that we didn't really care about with regards to VTuber research that we could reveal publicly before it's fully baked, it would be interesting to start off with the same thesis and then see how we differ in our writing. That could be a fun exercise. The varying tools of thought across the disciplines. Yeah, exactly. Because in talking about the research hell stuff with Mylan, which by the way, we still need to do that collab one day. I wonder how many people in V-Theorem we can rope into it. I think it'd be really fun. In that collab, I got a sense for how differently Mylan and I think about what makes for good research. And the fact that we differed, not that we disagreed, right? Because once I heard Mylan's point of view, I was like, okay, that makes absolute sense. But it was interesting to hear that we had both been trained to think of research differently. And I think writing will also, our different writing practices will also, I think, illuminate a lot of potentially individual differences. Maybe we don't want to overgeneralize to our fields, but potentially individual and field differences as well. It could be pretty interesting. I'm going to be writing a paper on the 3-episode rule coming up. If you want in, I'm going to need help with a data entry. Ooh, that would be very, very fun. I don't want to spoil too much about that, but that is very interesting because I do... I am aware of the 3-episode rule, and I'm curious as to what your angle is going to be for that. I actually heard of something similar happening here in Brazil long ago. That's insane to me. India, it's very unfortunate that this is happening lately. It's considered the norm that in the beginning you will give up some papers until your PhD is done. That's crazy. That's insane, because I feel like you get the same accolades no matter what, in the US at least, right? Because if you have a accomplished first author student, then everyone's going to know who you are. Yeah, that's crazy to me. I'm so sorry about that. It's mostly for chem and psych, and some other sciences have less trouble since a bit newer. Oh, interesting. Yeah, I'm sorry. That must be so terrible to deal with. I unfortunately can't recommend coming to the US for research, but if/when our research environment starts to lighten up, I mean, it's quite good in the US, I'll put it that way. Or historically, research in the US has been quite good. I think that is accurate to say. Relative to other places, right? Like if you choose a life of research, it's going to be hard no matter what. Researchers do not make a lot of money, but relatively the US has historically been quite good. Yeah. Interesting stuff. Yeah, I think part of why I love thinking about rhetoric is the kind of before and after of my own writing, where I feel like after I got the crap kicked out of me by my PI, all the walls of red letters and blood written all over my grant proposals, that like after taking those lessons to heart, I kind of looked back at my old writing and realized that like, gosh, it's bad. And my way of thinking was also quite bad. The benefit of graduate school, I think, is developing clarity of thought, which if you don't have that experience, like I don't know if anyone is actively aware of how clearly they're thinking, because you know, it's just impossible to tell, right? Like prior to graduate school, I never had the opinion that I was like bad at thinking, right? Like that's such an absurd thing to think. But then after graduate school, I was like, oh, there are more efficient ways to think about tackling problems than I was trained to do before graduate school. And it took a lot of years, I think, to refine that thinking tool. And writing is one style of problem solving, right? The problem being clear and effective communication. And also writing better also means that you can proofread your own logical thought as well, because if your writing doesn't make any sense, it could be a symptom that your thinking also doesn't make any sense. And it's really hard to be aware when your thinking does not make any sense unless you write better, right? Unless you put down your ideas and on paper and have someone else read it. If they understand, it's like mind transfer, right? Like if you can transfer your thoughts through words to somebody else and they understand, that means your thinking is good. If you cannot do that, it is likely that your thinking is bad, or the skill of writing that you have is also bad. If you know that you can write well, but you cannot communicate your ideas, it means that you're not thinking very well. So that's why I think the connection between writing and thinking is so important. It's not just other people need to be able to understand you. It's that if other people don't understand you, you probably don't know the problem as well as you think you do. And I think that's interesting. Clarity of thought is a difficult thing to point out the anatomy of. I think it really is difficult to point out, but I think that I mean, maybe for a high effort stream, I could write a research project, first draft and second draft, and then just show you. I kind of did in my flu video where I kind of showed like a crappy intro that I didn't like, and then I refined the intro to make it better. And I think I might do that. I might do it as a way to showcase the process of revision, right? Like maybe I'll take a viewer suggestion where you want me to tackle a particular problem in Alzheimer's disease, and I will come up with a research project. I won't actually make sure it's novel because I don't have the time. Coming up with something new is really hard, but I'll come up with something and I'll take a crack at it, and then maybe I'll revise it live. That might be kind of interesting to show you the difference between the first conception of a scientific idea and followed by the refinement, because the refinement I think is really satisfying. It's kind of like a Zen garden, right? Like the first copy of your idea is like dog water, and then you like even out the sand, and there's a level of peace that you get from reading an idea that makes sense versus not make any sense, right? I think that would be cool. Meson pi research, maybe something within my field. Maybe something in my field. Yeah, that would be very appreciated. I think it'd be cool, honestly. That sounds like a very fun stream idea. Describing how and why you would make certain edits would be very useful. I think it would be very useful. And like, am I the best science writer ever? I don't think so, but I think that I've always enjoyed writing, and actually you can see hints of the lack of clarity in my thought from high school. Just I never realized how muddled the clarity. Okay, I don't want to brag, right? Like I don't know how much more clearly I can be thinking, right? But I do know that I think much more clearly now than I ever did before. Like even though I'm not 25 anymore, right? I'm not like the peak of my cognitive speed. But in terms of experience, I'm much more efficient in thinking than I ever before in my entire life, which should give people hope, right? Like as you get older, you don't turn 25 and just get worse over time. You accumulate experience, which is just a different part of making you better. But in high school, like every English assignment that I had, I would write up a draft of like, didn't matter what English class it was, American literature, European literature, classics, narrative writing, whatever class I was taking, I would write an assignment, I would edit the assignment, and then the night before the deadline, I would throw the whole thing out and start over again every single time because I was embarrassed to be submitting work of that particular quality, and that often meant I got penalized for it because my second draft couldn't be edited. It's hard to even call it a second draft. It's more like just a second attempt at the assignment as a whole. And I think the signs that I was not thinking very clearly was the fact that I had to restart over and over again. Whereas now that I write for Python Neutrophil, right, the scripts that I have for these videos, I typically don't have to throw the whole thing out and do it from scratch again. I mean, except for the flu video. The flu video, I scrapped a lot of it because the flu was very confusing. And I was not thinking very clearly because the flu was so confusing, it's impossible to think clearly about it. But for a lot of my current scripts, like, what should give hope to people if you are struggling with writing is that I no longer have to revise like crazy. I mean, I still have to revise a lot, right? But in terms of the feeling of everything I wrote is garbage and I need to start over, that doesn't happen to me as much anymore, which I'm very grateful for because it's a huge waste of time. Huge waste of time. Do you think it's a failure of school level education that this level of clarity of thought isn't taught? I think it's really hard to do so because of a couple of different factors. One, you need to be able to have the basic tools in order to problem solve. And there's just not like a great way to do that up until you acquire a level of background that is important, right? Or useful in a way, right? Like you can't solve an engineering problem without knowing a little bit about engineering, unless you're really brilliant, right? Unless you're a genius kid. So for example, say this, you're in an engineering 101 class and your task is to build a robust bridge over 2 meters, between 2 meters of space between two desks. Sure, you might have the odd genius kid that has an inherent knowledge of engineering and can come up with a engineering design that solves it, but most people are not going to be that tuned in with engineering principles. And so you have to learn enough principles before you can actually start doing things. So why do I think that this is really hard to do at a pre-college level? Well, because you just got to learn your tools before you set out to actually do things, right? Like, in order to be a citizen, a well-educated citizen, I'm only going to speak about biology here because that's my expertise. Like, you should know basic anatomy, right? You should know basically how the body functions. You should know the systems by name, right? You should know, that there's a respiratory system, a digestive tract. You should know how the reproductive organs function. You should know how sex cells are created. You should know about the diversity of sexes. You should know a little bit about the menstrual cycle. Like that's not because you're gonna be a doctor, right? That's just being a citizen of the world, right? You should not graduate high school without knowing a little bit about what a period is. Both women and men, right? Everybody. Doesn't have to be sex. Everybody should know how the menstrual cycle works. That's just something you should know. And your education up until college is making sure that you understand those basic things. Now, is there room for higher level thinking? Well, the problem with our current education system is that there's very little incentive to get that deep into anything, because there is a priority of knowing things over knowing how to think. And do I think that that is necessarily a bad thing? I don't actually know. I flip-flop on this all the time because if you look at this is so messed up, but like some people just don't care about education, right? Some parents don't care about education. Some students, I mean, a lot of students don't care about education. A lot of parents don't care about education, right? A lot of people are not invested in the pursuit of refining brain things, right? They're not invested in that. And so what is the point of structuring a class where you are teaching deeper critical thinking if nobody cares? Teachers, students, parents, at least in the US. I've heard, from my streams from international, like I'm so US centric, from people that are from all over the world, they actually do have critical thinking baked into their education systems. And I guess I'm a little with the current level of money going into US education, I'm a little worried that there's not that incentive for students to care, because like it's intellectually like pretty low level to repeat things that you saw in class, right? And that's really unfortunate for students who want to excel. But the thing is that like for students who want to excel, if they work hard, if they get a scholarship, if if they excel at whatever higher education level that they can attain, they will do well because they're intrinsically motivated. Teaching how to critically think on a mass population scale, I think, is tough. I'm not saying it's not worthwhile. I'm just thinking that I get why it's not happening as much, I would say. Yeah. What about discovering that something doesn't exist? It's about that time, isn't it? Didn't that study about development stopping at 25 stop collecting data at age 25 or something? That one is extremely flawed, and I cite it not as like hard fact, but just as like common conception of your mental faculties degrading after a certain age. It doesn't. Even if it did, you still gain experience, which is invaluable. I want to make sure I put it that way. Krebs cycle. Why does it keep on getting longer? I have no idea what it is. I just saw the memes. The Krebs cycle is very complicated, although it's not even that complicated. It's complicated because it's one of the first biochemical pathways you learn of, but it's honestly not that bad relative to the immune system. But it is a metabolic pathway that happens in the process of converting sugar into energy, and the first time you encounter it, it's kind of crazy. You should just Google it to look at it. It's complicated, but med students are probably memeing on it because they have to learn from medical school, and it can get quite complicated depending on what you want to do with the outputs. Yeah, Mr. Krebs cycle. True. Yeah. Hot take, Krebs or citric acid cycle? I don't know if Krebs was a good person or not, because I don't know anything about the person Krebs, but I always feel like sometimes you run into a scientist who was like also terrible. So I'm actually a fan of the citric acid cycle. I think that is at least more functional than the Krebs cycle in a naming convention, because then citric acid is there. That means that's one less thing that you have to memorize about the citric acid cycle. Krebs is just easier to say, and it is what I was taught in high school, but by the time I was an undergrad, we were calling it the citric acid cycle. Yeah. Yeah, because I had this idea back when I was in graduate school and thinking about teaching high school. Like, if I were to teach critical thinking, I don't think I could do it unless it was wrapped into an AP biology class. And an AP biology class has to be prioritized to the AP exam, so there wouldn't even be enough time in there. What I think would be interesting to implement at a high school level is like science critique, and this could be open for anybody, where we take a like nutrition paper that is very easy to understand, right? Like, nutrition papers are not hard to comprehend. They're hard to make conclusions about, right? Like, they are interesting and very nuanced in how they intersect with health and biology. But in terms of just reading the words, nutrition is pretty easy. So we would read nutrition papers and we would do journal clubs. We would go line by line through the science paper and we would debate, because critical thinking is all about debate. It is about pressure testing your clarity of thought, and to do so in a concrete way, because you can do this in philosophy too. But I think the problem with critical thinking in philosophy is that it is very heady, and it does not have the appearance of something that is very practical. Because yeah, in philosophy you learn formal logic, and that's useful, I guess, if you like thinking that way. Like, if you are a mathematician, if you're a physicist, if you are a first principles first kind of guy, I could see that working for you. Like, a physicist taking philosophy makes complete sense to me, because physicists tend to be more about first principles. However, I think the number of people who are first principles type people are very low. I think as a proportion of the population, of course, I don't have data that backs this up, but I would say that most people do not have good at math phenotype. I put it that way. Now, whether or not that is because of a failure of education or some intrinsic difference in talent, I don't know. But I will say that practically, I think the formal logic route of teaching how to think is not good. I think what would be better is something that matters more to people intrinsically and is more concrete as a debate subject. So like we can read, like week 1, I thought about maybe let's read together a study that shows coffee consumption reduces the incidence of diabetes, or rather the consumption of coffee is correlated to the reduction in diabetes. And so that would be interesting because that's a very easy experimental setup. And what's difficult is what can you take away and what can't you take away? And I think having classes like that would be great. I think that could be a way you could do it. Unfortunately, I don't know if we could fit that in a high school curriculum in the United States. Although if you are from a country that cares a little more about high school education, maybe you could do it there. Pressure testing your clarity of thought. Heck yeah. Yeah, exactly. I think that's my favorite part of my current job. And I want, like, that's the part that I'm really, really going to miss about my job. And like streaming can kind of make up for that. But not quite in the same way, which is why I started this like cell tank idea where I would like to pressure test whatever ideas you have, as long as it's in the field of biology. You know, of course, the closer it is to my specialty, the better I'll be able to pressure test it. But I think it would be fun, like legitimately fun to pressure test ideas live on stage, because yeah, it's like so much of higher education is that back and forth. Like I joked about, I won't reveal it. There's something that has been in the ether with regards to a collab that I want to do where I could if there are biology VTubers out there who want their ideas pressure tested live, that would be very, very fun, I think. It's just right now, I mean, there may be biology VTubers out there who would be interested, but it's a lot to commit to, right? Like, not only does the biology VTuber have to have time, but they also have to have an idea that they want to pressure test, an idea that they want to pressure test and keep for themselves in case they want to do it in graduate school. If they have a free idea that they want to pressure test, like that'd be great. You know, that'd be a lot of fun. But the chance that happens, I think, is not zero, but pretty low, I think. I graduated high school 1 year before they started having philosophy classes. Yeah, philosophy, I think I should have taken it not because I would have found it very useful, but because I think I would have had fun. In retrospect, I was a little afraid because I know that the text can be quite challenging, especially challenging now on a generation of ChatGPT. But I think I still would have, like, if I were to ever go back to classes, say I'm retired, I made $1 billion off of Philo Neutrophil, and I retire, I would love to take a philosophy class in my old age. I think that would be great. This is a chatting stream. Yeah, I mean, I've missed y'all, and I'm almost done with my job, so it's nice to be able to talk a little bit, I think. A journal club, but it's a science communicator testing how well they analyze a paper you assign. I mean, Clockwork, if you want to throw your body to the wolves, if you want to read a paper and have me critique you live, I'll pressure you if you want that scrutiny. What's a pressure test? So it's just a general term for having your ideas challenged by other people, right? So I took the term from RTSs, like real-time strategy games, where, you know, say you're playing StarCraft and you want to follow a particular strategy. Well, the strategy might sound great on paper, but until you start playing with people with that strategy, it won't be properly pressure tested. And so without the pressure testing, you'll never know if your idea is actually good or not. So the pressure test is seeing whether or not your thoughts, your ideas are actually good instead of just good on paper. I saw your idea about Cell Tank. What is your plan for how to do that stream? I think it would be the viewer would submit some slides and a script for me to read out loud. I think an important kind of parasocial barrier for me is that I should essentially never hear your voice. I think that's a very strong line to separate creator from supporter that I would like to keep. But giving me a script that you would like for me to read, I think that would be the way to go about it. Yeah, and then I would give feedback, or I would point out ways in which your idea does not align well with what I know about biology. What do I do in my free time? It's a good question. Like, while I was employed, I would be doing this in my free time. But now that this will be my full-time job, it's a good question. I don't have an answer for you right now because I'm heading into untread ground. Pretend I typed this in all caps. I cannot express enough how down I am for that. Yeah, we can all watch Clockwork get grilled live. That would be really fun. Like I could pick you a paper, and I think what would be good actually is what my PI did for me when I was in graduate school. Is I would give you a paper, and then you prepare. I mean, this is asking so much of you, Clockwork, because this is asking you to do what I do essentially, which is, I don't know how comfortable that would be for you, but if you wanted to do like I assign you a paper and then you put together a full journal club, you explain every figure, and then you present it in front of an audience and me, then we could talk about ways in which I agree with you and ways in which there are ways that your understanding could be pressured, and we could find out that you're just goated, and like we could just find that I just like no extra comments. But I genuinely think that would be really, really fun for the audience to see like two biologists. And again, like it has to be a paper that I'm going to be more familiar with than you, because like there are areas in biology where you are much more the expert than I am, where it would not be productive for me to lean into your expertise, but somewhere in the middle I think would be appropriate. And that would probably mean something to do with cells. Like, you're not just a molecular biologist. Like, you are primarily in the field of protein and molecules, but it's not like you've never heard of other things. So picking a paper that is molecular, but also in the context of a cell, I think would be a pretty good space to pressure test your critical paper reading. That's not to say like Clockwork did all his own research for the flu segment that he had made for my video, and I think all that was done very well. But it's like the like reading a paper for comprehension and reading a paper for like critical analysis are two different modes of thinking, and I've never like, I don't know, Clockwork, if you were reading for critical analysis, but the only thing that you had to do to make the flu segment was to read for comprehension. And so to see you in an environment where you are reading critically, I think that would be cool. That would be dope. This is what I do anyway. If I'm going to force myself on the public, both the public and I should be aware of my shortcomings. We only get better together. That's a very nice sentiment. And I do think that is like a pretty unique appeal. And I also think it's like, I keep on referring to Naruto, but I think that like part of what makes content good for me is like the narrative behind everything, right? Like, I don't know, you can think of this as like Clockwork's like Chunin exam, right, from Naruto, right? This is the Chunin exam where there's an examiner, and you know, Clockwork could ace it, or there could be some drama with misreading something, or thinking too narrowly, or not thinking broadly enough, or taking the authors at their word, or maybe going too overboard. Because I find that like the problem that I had in graduate school is that like when I first gotten the ability, like when I first leveled up into critical thinking, as opposed to being like a memory machine, right? Because biology before graduate school was just memorizing. It was just memory machining with a little bit of critical analysis at the end of some classes, but most of the time it was just reading to understand. And when I leveled up into critical thinking in graduate school, I would take that critical thinking hammer and I would blow papers apart with it. I would be so cynical, I would be so skeptical that I wouldn't trust any paper. Every paper was never good enough. And my PI actually had to be like, bruh, there are limitations to techniques, and what makes for a good scientist is compartmentalizing the weaknesses of a paper to the strengths of the paper. So this is kind of like why I want to avoid the over skepticism of AlphaFold. And I know that my chat, I've read the comments, even though I didn't say it, that like I know that you understand that AlphaFold has to be backed up by human testing, which is the line that I have been towing. But in terms of how I think we are better to communicate that sentiment is AlphaFold is amazing. It's really, really good. We need to work together with AlphaFold to maximize its potential. And that part, I think, instead of being like, and we're essentially saying the same thing, but you can see how a difference in rhetoric, haha, plug for rhetoric, like these two statements are equivalent. AlphaFold is amazing, but we need to work with AlphaFold is essentially the same thing as I don't trust AlphaFold on its own. And we need to do human confirmation. You can see how one statement confers optimism, and the other statement, even though functionally they're the same, right? At the end of the day, we're saying the same thing, but functionally, that second statement is a lot more cynical, right? It's more pessimistic. It's more like, I don't know about AlphaFold. When I want to be very clear about my stance is that my internal feeling is that I am wowed by AlphaFold, but we can never give up our human side of science because AlphaFold can never be perfect. But never being perfect is what everything is, right? We just can't have our expectation be that it's perfect. And I know chat understands this, but the way that we communicate our ideas is important for the emotional affect, right? I'm expressing a limitation on optimism, and by wording it slightly differently, you may be expressing pessimism or skepticism. Yeah. Random question that you can feel free to skip. Have you seen the ads for Z-Biotics that allegedly break down the alcohol byproducts to prevent hangovers? Is that possible or is it nonsense? I don't know anything about Z-Biotics, so I cannot say, although I feel like if it actually worked that I would have heard of it by now, but yeah. Maybe it does, maybe it doesn't. Just because I haven't heard of it doesn't mean it doesn't work, but I feel like if it did work, there would be a lot of buzz around it. What is the difference between someone who has taught themselves to university level of comprehension of a scientific subject and a person who went to university? That's an excellent question, Treb. The value of a university education is pressure testing, right? Is the challenge of getting told that you are wrong. So someone who has learned everything about biology in a vacuum, that's fine from a theoretical perspective. Like, I have no problem believing, for example, that someone who is homeschooled could have a very high level of understanding of how the cell works. What I would have trouble believing is whether or not they can innovate on what is currently known, because refining your level of thought requires challenge, and unless you go to a university, you might not be challenged enough to the point where you can refine your level of thinking, skepticism, design, etc., etc. So the university, and this is not the true for all, some universities are better than others. Your mileage may vary depending on what professors you have. But for me, as someone who is a deep, deep believer in higher education, right? Like, I love higher education. I have had the idea that I would love to be an academic for so long. So this is my bias, right? I'm letting you know that I am deeply pilled by the academic system for all of its flaws. Like, I could love something while acknowledging that it is flawed. But the real benefit is not being taught by the teachers, but by being challenged by the teachers, by being told that you are wrong, and by being told that you need to go back to the drawing board and come back with a better idea. All the questions I wanted to ask have been answered. It's 1:20 a.m. and I feel bad for myself, so I shall get some sleep now. Please do. Sleep is very good for you. Thank you for coming. Chat is awesome. Chat is indeed awesome. I asked because I have a medical condition that prevents me from getting to university, but I'm 100% a scientist at heart. I'm so sorry that you are experiencing medical issues that prevent you from getting to university. I'll say that it's so tough, right? Because in order to have the full university experience, you need a two-way connection. Like, you can take MIT OpenCourseWare right now. You can take an MIT class for free right now. Google MIT OpenCourseWare, and you can have the entire thing right in front of you. You can be taught by some of the best professors in the entire world. The thing that you really need to get beyond a level of comprehension and into critical thought is that person telling you no. And like, that's why I would like to have that be a part of my stream, right, where I could tell you no. Like, I think that that would be the thing to do, right? Like, now, could I get you to the point where you are a professional? No, I don't think so. I'm just one guy. The benefits of a university is that you have a whole faculty whose job it is to refine your mind into a tool that can be used to solve problems. I'll put it that way. So, whether or not you could do the same experience remotely is an interesting question, whether or not there are going to be universities that move into that direction of having admissions not require on-campus attendance. But yeah, okay, so the classes aren't useless, right? Like, you go to be taught, but if you are capable of self-teaching through remote learning, functionally, I think that that is the same. The benefit of university is that two-way connection. I love the MIT open course stuff, but yeah, I feel like it's not a true substitution if you don't have someone like checking your work and answering your questions. Yeah, checking your work, answering your questions, that's like a major reason why you have a human in the room that you pay tuition to. Although MIT announced that if you make under $200,000, that they will cover your entire tuition, which is bonkers, right? That is so good for so many people who are going to be refined into some of the best thinkers that we have. And part of that is the connection that they are going to have with faculty who are invested in their success. So if you are thinking about attending university, one of the things that you might want to get a sense for is how much the professors actually care about educating the next generation, not just through classes, but through mentorship too, because science is deeply human, and you need that back and forth, I think. Too bad I'm English then. I mean, the UK has great universities as well, and with all the things that are happening in the US, Germany is going to be, I think, a huge powerhouse, or rather, will I don't want to say will replace the United States. I don't want to be that doomer about everything, but in terms of exciting places to do biology research, I think Germany will be. I'm looking at you, Germany. I'm keeping an eye out on you. But yeah. Yeah. I think these are all very fun ideas and thoughts to discuss, especially because I'm in the mood to kind of reflect on my university experience. I actually know people that went to my graduate school from my class who are working in Germany right now, and I have to say that, like, the person I know who's in Germany that came from my graduate class, one of the smartest people I've ever known, one of the hardest working, smartest people I've ever known. Like, they are getting talent and investment, and it's going to be very exciting to watch that star continue to shine. Because I'm not saying Germany is up and coming; they've been strong. It's just that the U.S. has been the hub, the crown jewel of health and biology research. And so while Germany has always been a potent force in biology research, I'm excited to see what happens through the current state of the US. What should I call you? Phi is fine. Phi, Phylum, or even Phil. When I go watch other streamers and they say my name out loud, sometimes they'll call me Phil, which I think is very cute. My true passion is in paleontology, and it pains me that there's an undergrad course being taught by the guy who rediscovered Spinosaurus in 1014. Paleontology, that is a subject that I know very, very little of. My only exposure to anything to do with paleontology is my incidental readings of Darwin, who had needed to draw from the fossil record for his theory of evolution. So I think it's very, very cool to think about studying animals that have lived that long ago. Very, very, very neat. It took over a century, but Bismarck's dream is finally being realized. You know. Yeah, yeah. 1014, 2014. It's so funny because I didn't even realize that that was wrong. 1014, yes, that person would be a medical marvel if that person was still around teaching this class today. Yeah. I want to hear how classes have been going for those of you who are in classes. Because we're at the end of the semester. Has the finals been going well? Have you been stressed or pushed to the brink, or have things been kind of chill? I'm very grateful to never have to take another exam ever again, by the way, because while I'm a very good test taker, I feel like every high pressure exam I've had to take has taken years off of my life. um, yeah, I was a kid that never learned good study habits until graduate school. By the time I was in graduate school, it was kind of too late, right? The benefits that I would have gotten by having better study habits were limited by how late I had to adapt them. I'm trying to start school again for the first time in literally 15 years. That's very, very exciting. That is so exciting. That's very, very cool. Do you know how to hit about the school system? Please let me know. Speaking of Spinosaurids, when are they going to return in the Irritator? Oh, I don't know what that means. I wonder what that means. Finished my AP Bio exam, so things have been going great. Still waiting on the NYS Regents exam, though. Oh, very, very fun, Kevin. Although if you took your AP Bio exam, I'm sure you will have done well on the Regents, too. Very, very fun. Very cool. 90% of it is anatomy, and the other 90% is converting the anatomical traits to code to feed to a program that builds the evolutionary tree. Ooh, that's very, very fun. I do think that's very, very neat. I think anatomy, again, I shouted out anatomists in this prion discussion today because that is a part of biology that I cannot get into. To me, learning anatomy is like so visceral. Like you actually have to learn to look at bones and stuff and muscles. I don't know if you've seen this gif of like individual muscle fibers being electrically stimulated, and it freaks me out. That's why I stay in the realm of cells, because once they're cells, they don't really look that different than any other microorganisms. But once they connect into tissue and anatomy and stuff like that, I get very weirded out, which is why, why I would have made for a pretty bad doctor. I don't actually like anatomy. Every dissection course still haunts me. Yes, dissection. As a biologist, as a molecular biologist, I can say that, dissection was not very fun for me. I had to do a lot for my graduate research, unfortunately. And like, I've not fully recovered from having to do that. Like, I could not compartmentalize my beloved research animals with how much I adored rodents. So I'm glad to not be doing active research in that regard as well, right? Like, I never want to handle a mouse that I know is going to die. I don't regret doing any of the research. I'm glad that I'm not doing it anymore. But yeah. I actually, for those of you who don't know, I skipped my AP Bio exam. I was not ready for it when I was in high school because the volume of memorization that you need to do well on the AP Bio exam is so much, and my memory is very, very bad. So I will say that if you have a bad memory, there's still hope for you. You can still do biology. Again, because once you are working in biology, you do not have to have encyclopedic, cold recall knowledge of the biology systems, but you do need a working knowledge of the biology systems. So if you struggle with memorization, that doesn't mean you're a bad biologist. It means that you are probably not going to do very well in the AP exam. What other science areas do you wish you could have taken classes in or studied? I wish I had taken immunology or physiology. I took physiology and I did not do well enough to convince myself that I could be a doctor, so I don't regret it in the sense where it was very informative that I should not do medicine. I wish I had finished my intro to neurobiology class in undergraduate. I dropped it because I had too many things on my plate at that point. I stayed through enough of it to know that I would love to do neurobiology in the future. I just couldn't manage that many classes all at once. I wish I took immunology for sure. Like that is 100% a regret that I will forever have because I am learning immunology as I go, and maybe, you know what would be nice? Maybe I buy an immunology textbook and I go through chapter by chapter on stream, not reading the textbook, but summarizing it. Oh, that would be really good. That would be really good. I buy an immunology textbook and I force myself to learn all of immunology from the ground up. That would actually make my videos better because I am learning immunology as I need it. So I can't actually critically think in the field of immunology beyond what I currently know. But if I had the sandbag, training every day, punching a sandbag, and learning immuno that way, I could actually do a lot more critical thinking to have that background. That would be really good. I'm gonna write that down actually. Yeah, that would be awesome. Let's do it in general so I remember. I'm gonna buy an immunology textbook. I'm gonna buy an immunology textbook. That would be awesome. Yeah. What else? I wish that I took biophysics from a professor that cared. I had a biophysics professor that just wrote things on the board, just derived equations, which again, I think works if you're a principles first kind of learner, but as someone who is not a principles first learner, it was very, very challenging, even though I think I would love the field. Philosophy, again, I really wish I took philosophy just because I think it would be fun. I wish I took more rhetoric classes. I realized I loved rhetoric in my last semester of undergraduate, which was far too late. If I had known that I would find the craft of language so enticing, I would have taken a lot more English classes. Yeah. And it's practical, right? Because I use the skills that I learned about, or I use the principles of rhetoric in my own phytonutrifil scripts all the time. It's not. I wish I had learned more about it so that I could do it better. Math is taught as just problems and questions, but it should really be taught as a programming language. I using functions to code and not to just solve questions. So the US is trying that through a program called Common Core, with mixed results, I would say. So the new way to learn math has been designed by like a Harvard fancy pants PhD, many years in education from first principles. So kind of like the way that you are describing. I'm not a math person, so I can't rigorously assess to what degree I agree with that. But it's really hard to learn if you've never learned it before. And so like teachers are having all sorts of difficulties with it. Parents of children are having all sorts of difficulties with it. It's a little too early to determine whether or not the children who grew up on this more programming language, intuitive mathematics, problem solving focused math core have paid off in a meaningful way, but I'd be curious to follow up on that. I don't know the general consensus as to whether or not that ended up being a good thing, but it is being tried out in the United States. I wish I hadn't been so afraid of the 1D I got in algebra that I engineered my entire high school curriculum to max out chemistry and biology just so that I can avoid trig. That is very fair. I think calculus is a useful conceptual tool for biologists. And I'm surprised, Clockwork, because your knowledge of biochem is like pretty good. At least your knowledge of structural biology is pretty good. But I think the background of calculus combined with thermodynamics, I think, would make for a very robust understanding of biochemistry as a whole. Even though I don't think you need it, right? For YouTube, I don't think you need that robust understanding of calculus and thermodynamics. But if you wanted like omega biochemistry background, then I think calculus and biochemistry thermodynamics and calculus are not avoidable if you wanted that like omega deep background. Being taught, sorry, I'm catching up on chat. being taught as you learning tools to solve puzzles you pose yourself. Yeah, like reading textbook live. I think that's what that is referring to. So that is what I am going to do. But I would need recommendations from immuno textbook. Thought I could rote memorize my way to success in biology. I mean, I did too. Just because I was not good at it didn't mean I didn't bust my ass doing it for real. I struggled a lot. Like, I struggled a lot in biology because of the rote memorization, but I was very bad at it. So I struggled a lot with that as a study technique. First time I got a grade 0, our F equivalent was at college. It was a humbling experience. I say this with all the love in my heart. I don't know what system you're involved with, but if you are in the US, you should not get an F in any course. Not because they're easy, but because you could drop them. Please, from a teacher right now, if you are bombing a class, drop it. It's not a bad thing. It actually, I've dropped classes before too. Like, if you are actively bombing a course, if your university allows you to drop it, drop it. It is only a positive for you. And I still got to go to my dream graduate school, even though I've dropped classes I wasn't doing well in. It is for that forgiveness if you are in the US. What do you mean rhetoric classes? So rhetoric is the study of how to construct language for meaning, for purpose. That is the field of rhetoric. RHETORIC. Yeah, that CDM's got it. Thank you. I had taken more law electives if I had known how much I'd need them afterwards. That's interesting. I assume that's because engineering is often very intimately connected with ethics and social implications, and I assume a degree in a class in law would be quite useful for that, which is interesting, I think. An anecdote of how far ahead the US is in biomed research. I remember my oncologist explained it to my mom that it is the equivalent of the UK still using steam engines to the US using jet engines. Yeah. I mean, I don't know a lot about the UK biomed scene, but I know that there are famous universities in the UK. I admit I'm not that familiar, but yeah, I guess if I were to pick a country in Europe that I would go to for biomed research, it would probably be Germany. Yeah. Calculus is a very powerful tool of thought for modeling phenomena that aggregate over time. Yes, exactly, Marlon. Like, it is there, like the fundamental understanding of what a derivative and what an integral is. Those are, it's not like do I have to do it in my day-to-day as a biology teacher? Kind of. Like, there have been derivative stuff that you need to know in terms of how to model certain biochemical kinetics, but you don't have to write out the equation yourself. Oftentimes you just grab the equation online and plug in the parameters, but a working knowledge of thermodynamics requires the working knowledge of calculus. Yeah, there's a certain deadline after which you can no longer drop classes, which comes relatively early on. So in both my graduate school and the current school that I teach at, the deadline to drop is so late into the semester. I think there has been a push to procrastinate the deadline to drop because it enables students to have more forgiveness for, I almost said screwing things up, but that's kind of victim blamey way of putting it, which I don't agree with. It allows for more forgiveness in terms of, sometimes in your 4 years of university, something terrible happens, a non-zero percentage of the time, and dropping classes enables there to be a mechanism by which that is not bad for the student. And again, I got into my dream top 10 university for graduate school, bragging. I know, I do it all the time. And every time I do it after stream, I go, can you just shut up and not brag about it? But I want to brag about it. I want so badly to tell you where I've had my education, because I am insanely proud of where I've come from. But I won't because of the docs thing. But I say that this time, not just to brag, but also to remind people that dropping will not stop you from getting into a top university for graduate school. It just doesn't. So please do. So what kind of teacher were you? Sure, bio, but in which field? I taught a lab class that was focused on engineering biology. So not just how to answer questions in biology. So a basic biology researcher, which is what I was trained in, focuses on answering things that we don't know about biology. And engineering biology is making use of living organisms like, E. coli or yeast or something to solve problems like climate change, like cancer, like, whatever, malaria, tuberculosis, whatever you want. You deserve to brag? Well, yeah, the doxing, never mind. Yeah, proud good, docs bad, proud good, docs bad, brag fine. Yes. I think that is true, but I just feel that, that part of me that is like, I'm very okay with bragging, by the way. If you want to brag in chat, like I think humble bragging is the worst type of bragging. Humble bragging, I think, from my perspective, I know people don't like bragging in general, but like the worst kind of bragging is the humble brag. That is, or the subtle brag, or bragging about someone else and taking ownership of their success inappropriately, right? Like as a teacher, do I do it? Yes, to a certain degree, right? Like I can look at a student who has accolades and go, I played a small role in that student's success. But that's me taking the appropriate amount of ownership, right? I played a small role in that person's success. It's taking ownership of someone's success through other means that is frustrating. But the worst brag is the humble brag, where I've had I've had classmates do this a lot, which has always been so infuriating. I've told the story on stream before, but there was a case where I was overhearing a classmate of mine confront someone because they had both done, in their eyes, very poorly on this engineering exam. And so there was someone who was crying on the other side of the phone who I could hear because this person was playing this conversation on speakerphone for some stupid reason, in a public space, by the way, where I was there, right? And this classmate of mine, I had heard from the speakerphone that there was someone who was crying who had gotten a 54 on an engineering exam, and that's devastating, right? It's freshman year, it's early on in our education, or sophomore year, I think it was, very early in our education, and this guy was like, oh, I'm so sorry to hear that. I didn't do very well either. And then the guy revealed that he got like an 89. And there's no doubt in my mind that was done maliciously, right? No person with functioning and like functioning empathy, right, takes a look at someone who got a 50 whatever and is crying, and then shares that they had gotten 1 point away from an A and considers that bad at a university level. I could see an 8-year-old doing it because they don't know better, but you're a 20-year-old and you are saying, hold on, let me take this to the logical conclusion, an 89 is bad, so a 50 is the worst thing. That's bonkers, right? And that's very distasteful. There's also a gender dynamic between the two, where the humble bragging guy was a guy, the person who got a 50 was a girl. I can't confirm that there's a gender dynamic, but what I will say is that there's a phenotype of guy who puts down women like that. And it was very, very annoying to see. Very, very annoying to see. If you want to show off, just go all in, guys. Yes, and take ownership of the appropriate amount of ownership of what you are proud of, right? Like, so I'll say that, like, the right way to brag is to emphasize exactly what got you there. And what got me to my dream school was I busted my ass. I followed like my passion for teaching, which is really important because this top school loves basic science research and loves the like OG conception of university as being a place of higher learning and not like a place where you grind out jobs, right? Because that is becoming more and more of the university incentive. And I think they interpreted my very teaching heavy resume as being someone who is more of the traditional academic mindset. And then, of course, like my parents, right, very encouraging of all my academic pursuits, although they were sad that I didn't want to be a doctor no more. Yeah, and then all the professors that wrote me good letters and helped guide me along the way, where I never like take ownership of like that success is thinking that I'm intrinsically different than anybody else. Like I'm not built differently. I'm actually built worse. My bad memory means I'm actually built worse than a lot of people. It's just I've had a lot of help along the way, and I cannot emphasize how lucky that is. So the luck is that I've had a lot of help. The work is that I've busted my ass, a lot of near all-nighters and not giving up in the face of failure and dropping classes when I needed to drop classes and taking classes that would help me communicate how awesome of a candidate I am. I mean, that is real. The incentive of taking writing classes is that you get to brag in a way where people don't hate you because you learn how to communicate better and, you know, speak better for when you interview with people. So that's, I think, the right way to brag. And anybody can do it in my Discord. Anybody can do it in chat. We should all celebrate, right? That's why I have the victories channel in my Discord. Is for people to brag. Please brag. I want to hear about how awesome you are doing. And then there's the opposite side of that, where's the L posting, where sometimes things don't go very well. And that happens all the time as well, right? Like, I think it would have been funny if I got my job non-renewed in the L posting channel. Now that I think about it, that would be very funny. Yeah. Yeah. So please do some more bragging in that channel of the Discord. Am I on top chat or all messages? I actually don't know which one I'm on right now. I'm on top chat, I believe. Oh, excuse me. Yeah. Out of the three sciences, physics, biology, and chemistry, biology is 100% my strongest. Yeah, it was for me too, for I think obvious reasons, where I think biology, if you are someone who is more of a story learner, because I think like the more mathy subjects, quote unquote, the more hard sciences that are more based in from first principles, that requires a more logical thought. But biology requires more narrative thought. There is no first principle other than evolution. Evolution is your only first principle. Outside of evolution, you just have to know and learn how things work, which I think people from physics might find disgusting. I know I talked to Carbon, who is another VTuber who is a physicist, and we had talked briefly on his channel about our relative passion for our own field and our relative distaste for each other's fields, right? I don't like physics because it is based in first principles. Like, you do not get to appreciate physics, I think, in a real way until you are very good at math. And I'm not that good at math. I was good enough to pass my tests, but I feel like I did not have a deeper appreciation for math beyond that. Wow, you must be super smart, still has nightmares about failing classes 10 years later. Exactly. Like, I got a lot of smoke blown up my my membrane when I was young for being an above average student in class. And all that meant, I think when you are that young, I think it just means that, like, for a non-zero number of students, it just means that you don't have any outstanding behavioral issues. Which, you know, I'm not assigning blame or anything, right? But if you are a child and you are able to focus in class, behavioral issues or other issues, right? I'm not assigning blame or faulting it for anybody, but like, when you are that young, some things have an outsized impact on how well you learn in class. And I was a kid that did not have that kind of impediment to my ability to learn. So at a very young age, because I was like a pretty normal kid with parents who very valued education and pushed me, I did, like, you know, better than average. Whereas like other students didn't really have, you know, either they didn't have parents that cared as much about how well they were doing in class, or for some reason or another, you know, not going to say what exactly, because I wouldn't have even known at the time, but you know, they just couldn't focus or didn't want to learn or didn't want to behave or didn't care, right? And just for being above average, I had, you know, a lot of praise thrown on me as a kid, which did bad things to me in terms of assigning intelligence to a component of my being, my personhood, which was ultimately very harmful. When anytime anything made me feel dumb, I like crashed out. So I had to unpack all that in therapy, which I've talked about before. So if you're raising kids, it's not good to say that they're smart. I do believe that. It was good to have the confidence. It was good to hear that I was smart as a kid, because I think that's empowering, right? Like, I can do it because I'm smart. But as an adult, it's like, well, if I can't do something, that must mean I'm dumb. And that's bad. I think as a kid, it's much more valuable to hear that you can do something because you want it enough and that you are working hard enough to get it. And I think that's what the parenting books these days say nowadays anyway. Yeah. Yeah. I did a cool youth science project and got second place in the region despite killing all of my bacteria. I mean, the thing is, and I had a conversation with Tox that was very interesting about failures in biology labs, which I think is very interesting. And it kind of gets to the point where, Mr. Winter, you're not alone in having all of your bacteria die. That happens all the time in biology. Like, if you take biology lab and chemistry lab, you'll find that the function of both are actually very different, because chemistry lab, if you're bad at like purifying a thing, your yield is very low, you might get penalized, right? And that's just skill issue. But in biology lab, there's a very real chance that you did everything according to instructions and it still doesn't work, because biology is messy. And that's not because you didn't do anything wrong. You could have done something wrong. You may have done something wrong. You probably did something wrong, but there's only so many words that you can fit in a protocol, right? There's only so many things that you can convey in words to a student, and they could screw up in ways you could never predict, right? And so if you have a step where your semester-long project requires your bacteria to grow and they die, well then, do you just fail the rest of the class? I think in biology labs it's much more forgiving. It's my job as the biology teacher to redo your whole thing to make sure that you can learn. And so chemistry doesn't really afford you that luxury. You fail to purify a product, you fail to synthesize a product, you just fail. Or you get penalized. You don't fail, but you get penalized for it. So biology, there's so much failure involved in it that I do completely agree that even a project where you have all of your bacteria die can still be worthy of second place. It is very, very interesting. Sometimes I wonder if those talented and gifted programs do more harm than good in the long run. I think it depends on maybe how you package them, because I was a part of some accelerated programs as a kid, and selfishly that benefited me because that kept me in a separate class from students that were violent. Right? It's harder to learn when you are worried about your own physical safety. And, there's a very tough philosophical conundrum that happens here, where if you separate the kids who are doing well academically from the kids who are not doing well academically, are you just dooming the kids who are not doing well academically to a life where they cannot succeed? But if you mingle the two classes, are you hamstringing the students who are doing well by putting them in an environment where they are potentially unsafe? I don't have an answer for you. What I will say is that I benefited a lot from being physically separated by class from students that were, well, we'll say rowdier in our school system, put it that way. A lot more non-learning happening in those classes. And again, that's why I'm trying to be very careful with my language, because I don't want to blame the kids because they're kids, but something in their lives has caused them to not care about education, and I don't have answers for how to change that. It is tough. But I benefited, I'll say that. Not only did it put a ton of pressure on me, they also missed my obvious signs of ADHD and possibly other neurodivergent conditions. Yeah, I'll say that not everybody benefits from that kind of separation. Very yes. I felt like being good at biology in particular was the only thing that made me a good person. So when I started pushing up against more complex topics, I crashed out really hard. Oh yeah. I had that with medicine. I felt for a long time that my desire to be a doctor, a medical professional, was what made me a good person. When that's a very myopic way of morality, but as like a dumb boy, I'll say that in particular because Like, I don't have data, but I think that there is a problem with how I was conditioned as a boy, or a cell from a male body, where morality is often impressed upon as being more black and white, where I thought the only way that I can be a good person is by devoting my ability to do biology and apply it in a way that is helpful for people. I'm not saying that doctors aren't good people, right? But there are more ways to be good or to contribute than just doing medicine. Oh, I didn't feed them for weeks, poor guys. That would do it, actually. Once I killed my yeast cultures and I cried myself to sleep, like losing a pet. Now, to be fair, I was very hormonal too. That is fair. I've actually had students, like, it's so funny how jaded, not jaded, but like the more biology you do in the lab, the more you get desensitized to certain things. And I'm often reminded as to how shocking it is the first time a biology student loses a culture. So I'll say, Pedro, that you are not the only one that has had that happen. I've seen it with my own eyes. They didn't cry in front of me, but they did take it pretty hard. And so we were growing like mammalian cells, right? Like human embryonic kidney cells. And when we were done with them, you have to bleach them, right? You have to kill them. You can't just throw them in the biohazard bin because they contain human tissue, or mammalian tissue that could be vectors for disease, right? So you need to eliminate them for safety reasons. And so I offhandedly mentioned that that the cells would be bleached, and the gasps from the students were very audible. And I was like, why is that crazy? That's not crazy. And the same thing happened when we were doing like E. coli culture, where you have to, in order to prevent DNA sequences that can confer antibiotic resistance from leaching out into the environment, you have to bleach your bacteria. And the cries that I heard from the class were surprising and very endearing. I'll put it that way. Very endearing. Hi-Fi, where can one get petri dishes? I'd like to cultivate bacteria. Don't do it. If you do not know what you are doing, you could potentially really hurt yourself and the people around you. You should only be cultivating bacteria in a lab that is ranked for biosafety. You do not know what you will be culturing, right? For example, if you culture something, so we used to do this like for outreach in our graduate school program, where we would teach the public about the different kinds of bacteria, good bacteria, bad bacteria, how dirty things can be. But the thing is that those were cultured in a very safe environment where there could be no contamination into the environment or exposure to real people. Because if your argument is that there are harmful bacteria in things like hand dryers, and then you are careless with your culturing, you could potentially be culturing harmful bacteria, and harmful bacteria can sporulate, right? You can get some transmission through the air. Is it common for bacteria? I don't really think so, but it is a possibility. So we did it in our cultured conditions, and then when we showed the plates to the public, we did not allow them to open the plates. We kept them sealed shut so they would not be exposed to anything potentially harmful. So if you do not know what you are doing, if you have not had an experienced biologist train you in a certain technique, do not do this at home. I cannot stress that enough. That being said, I really wish I had been made aware of pathology as a career path and discipline. What a great area of study. I think we talked about this before, Clockwork. If I were to become a doctor, I think I would have chosen to become a pathologist. You don't have to talk to patients, and you get to look at things under a microscope. It sounds like a great area of medicine for me. Last time I saw someone lose a culture, they discovered antibiotics. A rarity. Most of the time you lose a culture, it is to contamination of some of the worst smelling and ugly looking things you've ever seen. I'm going to accidentally create a bioweapon or something, or some all-powerful homunculus one day. I mean, we're trying to do things to prevent that from happening. The one thing I've seen mentioned about gifted kids is they often suffer because nobody teaches them how to learn stuff before they cruise through the basic stuff, and they crash out when they can't. Yeah, exactly. Like, I think that is very common. I might talk about it more or again in a future stream, because one of the streams that I have planned for my streamathon, which is going to take place in 2 weeks, right? So in 2 weeks, I'm going to be streaming every day, talking about biology, talking about academia, talking about classes and stuff in a much deeper depth. So if you want to have this conversation again, I'm very happy to do it, because it is something that is so important to me in particular, as I was never called a gifted kid, is a funny thing. I was just shy of being gifted. I was sub-gifted, just accelerated, sub-gifted. But, you know, whether or not that matters is a different question. But yeah, like the ability to learn, you know, before you get to undergraduate, it is, if you have good memorization skills, it's pretty straightforward because you're not really being tested in those more critical thinky areas, right? The ability to make connections and conclusions and to hypothesize aren't really things that you get to do until you get to university, at least in the US. So if you are spec'd for good memory, like you really shouldn't be called gifted, right? Like it's just, you can memorize good. Although if you're good at math, that's like not really memory dependent. That's just you having good first principles. And then maybe I can make the argument for that being allowed to be called gifted. But I still think from a rhetoric point of view, gifted is maybe not the word we want to use anymore, although I don't really know what word we would use instead, because I benefited from the segregation of, you know, keeping all the students who are doing well away from the students who are not doing well. I don't know. Like, as a teacher who has been fortunate enough to teach students who have always been very motivated, like even my summertime job, right? I took a job, my first job ever was to tutor for the SAT. I was a big nerd, right? Like, it's no surprising that Find the Neutrophil was good at school. I hope that's not a surprise. So I taught students how to ace the SAT. And, you know, their parents paid good money for that service. Of course, I was paid minimum wage, but they paid the tutoring center a lot of money. And so there was investment from these children because they were, I'll say, culturally motivated to do well here, to have parents whose expectations were set that they would spend money for them to get into high, like top tier universities. So I've never really had to teach students that were not motivated. And that is where I feel very deeply for public school teachers, right? Like, I could not imagine myself teaching at that level, because having to be both a teacher and a therapist and a behavioral counselor sounds stupid. Not stupid as in not worth doing, but something that I could not do effectively, right? Like, I don't have the emotional intelligence to be someone's therapist and teacher. I want to talk about biology. I don't want to talk about your home life. and it's so unfortunate that, like, that balance is so disrupted in so many students, right? Like, I wish, like, I have these like I've had these like Mr. Beast aspirations, right? Like, if I were Mr. Beast and I were to actually put my money into a cause, not research, but a cause, right? A philanthropic program, I think the idea that I had been playing with, and again, I don't know if this is problematic or if this is a stupid idea, but something that I would have loved to fund if I had Mr. Beast money was something like a free nursing program, a program that would take in applicants from backgrounds that are very troubled and pay entirely for their room and board and give them training for a nursing degree. Because if you are sufficiently motivated and can learn and like want to like crawl out of circumstances, I think that that is one potential path that could be done. And, you know, nursing is a good job. It is a good career. It is tough. It is not for the faint of heart. It is not easy. But this kind of program would reward people who are like motivated enough and have the means to, or like the means part would be taken care of, like free room and board, essentially a full way scholarship for a nursing program, where this is a way to have like a way to make a living without it being a college thing, right? Because university, as much as I love university, it is not straightforward. Once you graduate from a degree, it is not an easy path to having a job. You could learn whatever in university. Some degrees are better at making return than others, but a nursing degree, that will earn you a quality of life that is pretty good. And so, again, I don't know, chat, you can tell me if that's dumb, but if I had Mr. Beast money, that's what I would want to do with it, I think. Let's say if I wanted a science communicator, someone like yourself, to give a virtual lecture on my campus, is it possible? How could I do that? Oh my God, I would love to do an actual virtual lecture. And there are guest speakers that happen all the time from outside of academia to universities, right? Like, there's like there's a comedian. I won't like give away who it is just because this is connected to some troubling things, but it is a comedian that analyzes conspiracy theories, and this comedian has no background in academia, but is doing university talks because of his real lived experience as someone who researches conspiracy theories out in the open. So there are people that can come in from outside of your university. As for how to do that, if you are a student, it might be good to just ask a faculty member that you trust to help set that up for you, because it would probably be like a departmental thing, and you know, I'll do it for free. You don't have to pay me. So if you want me to talk at your university, I have an email address that's in my YouTube description. If you are sending me an email from a .edu account from a particular university, that's going to give you a lot of credibility. But I don't know what I would talk about. Maybe science communication as a field. That would be kind of fun. If you have something in mind that you would like for me to talk about, but like, I love the university system. So yeah, I would be down to talk at universities. It's up to you to make that happen though. That'd be really cool. We're not taught how to fail, and there are so many failures in science. How to live with it and try again. Exactly. That is not for the faint of heart as well, because it is very stressful. Prakiji cameo, yeah, she did meow. It is pretty close to her mealtime. I will call a stream soon. Yeah, right when I have to go feed Prakiji, but I just miss talking to you all, and I'm feeling very chatty. This guy is right. We don't reward nurses and always have a shortage of them. Yeah, and I think that an entry point into this very tough field, this very important field, is very important field that requires a lot of hard work and discipline. But like, in terms of like why I think this is such a good like Mr. Beast kind of idea, is like, it is not a giveaway, right? It is incentive, where you're incentivizing people who have very little opportunity afforded to them otherwise to be able to get out from where they are, right? This is like American dream stuff, right? Like, I have always felt so privileged in the fact that I never had to think about where my next meal was coming from. I never had to think about whether or not my parents approved of my lifestyle or my career goals, although again, they were disappointed that I didn't want to be a medical doctor. But what parent wouldn't, in terms of a medical profession is robust to recession. It is robust to so many things. It is very well earning. I just couldn't convince myself to do it. So I understand why they would be sad about that. But I never had to doubt whether or not my parents believed in me. Like, I think they believe in me more than I believe in myself, actually, which is not something that everybody has. So, you know, a scholarship program for a nursing degree, I don't know if that's practically a smart idea, but to me, it solves a couple of very important problems all at once. Now, that is to say that like, not everybody can be a nurse, right? Like, I think there should be an application process where you screen for people who are hard enough to become nurses. Like, I would be the worst nurse in the world, right? Like, I'm a squishy, sensitive kind of guy. Like, I am weak and not at all strong in any kind of way. I am very much like an academic in that regard, where I am in my ivory tower, and I am doing science communication for people who are not in the ivory tower. But in terms of like, do I want to be out in the front lines of helping people? I kind of don't. I'll talk at a university. I love that as an idea, and I'll talk to all of you, and I'll make videos to explain biology and stuff. But in terms of like screening for people who are like careful enough to be a nurse, pay attention enough to be a nurse, are determined and willing and focused enough to become nurses, I think, you know, that would be incredible. Because I've actually worked with people that come from troubled backgrounds, and they do work as like lab technicians, for example, and there are they have skills that they bring in that I don't really have, right? A lab technician, for example, managing a mouse colony, that requires a level of attention to detail and scheduling management that I do not have, because I just don't have those skills. Like my background, like if I had to rank my brain is in terms of my stats, right? The highest stat that my brain has is a mixture of curiosity and fixation and storytelling, right? Like my starting bonus, I don't know, you put it that way in gaming terms, is curiosity and storytelling. That is, I think, I'm not going to call myself smart, but that is what I am best at. And people that are better at organization, at discipline, they have the skills to do lab colony maintenance better than I do, because I am kind of messy in my discipline and in my ability to keep a colony going. And I remember working with someone who went to like a really rough school, like from, a background that was not nearly as privileged as mine was. And I was like, I bet that this person would make actually for a much better nurse than I would, because when you gave them a task, they did it perfectly every time. And in a way that I could not imagine myself doing the same. Right. And it shows that like society rewards kind of different skills differently, which is unfortunate, right? But what is true is that, different people come to the table with different skills. Mine just happened to be more aligned with more heady, abstract things. But in terms of like the skills that it takes to be a strong person, enough strength to actually do nursing, it wouldn't be a program that just would take anybody from a place with hard means, but a place from people who do not have the means to put themselves through college, but also have the guts and the hard work to actually be a nurse. I think that'd be great. And I think part of that is probably just guilt of being like, I had it pretty good. Nursing staff is like the muscles of the health sector. Nothing works without them. Exactly. Like, it is a very valuable job for the health system. It is a very valuable job for the patient, because the patient, the nurse is the one that's going to be interacting with the patient a lot of the time. They're the front lines and ears of the whole system. So, and the system does reward that in a way, obviously not as much as doctors, but in terms of peeling out of a life of, that's hard. I think it could be a pretty interesting route outside, which means, continue to watch my videos. That plays well with the algorithm. Maybe I will have Mr. Beast money one day, or maybe I will convince enough people to pull together money for this scholarship to actually happen, right? Like I don't even know if nursing programs have on-site dormitories. I don't think they do, but that's a dream, I guess you could say, like a very real, not one that I'm thinking very seriously about attaining, because what is my actual dream that I would love to attain? It's this right here, baby. It's doing this full time. It is and there will be a process to make sure that I can support myself and put food in my mouth based off of this as my job. But I have confidence that eventually I will make it there. But in terms of the unattainable pie in the sky dream, that would be to have this like program where, you take people who are looking for a way out of a hard life and enabling them to do the best that they can do, which I think is interesting and a cool idea. Becky, we need you in that tower. Thank you for believing that is so. There's so much sort of anti-ivory tower rhetoric nowadays, especially in the US where I'm based in, that it's not that the ivory tower itself is bad. It's just that connection to the ivory tower is important, right? Like, people, if we do research with your tax dollars, should be well informed. Now, part of that is the media ecosystem, right? Like, we need like a generation of young scientists to take advantage of TikTok and stuff to make it very clear to the average American what their tax dollars are going towards, because I feel like that has been a major failing of science communicators. And I say this as a science communicator, and I'll say this as a science communicator who understands that I'm not doing it right either, right? Like, if I really prioritize conveying my information to the broadest audience possible, I would be on TikTok, because I think you in my chat, you are more scientifically inclined, I think, than the average TikTok user. You have found your way to me either through the virtual YouTuber subreddit or through the algorithm, and something about my content, the deep dive on my content, resonated with you in some form or another, and then you showed up. TikTok is where you get discoverability, like virality, right? Like, that is where your stuff gets pushed to millions of people who are maybe not that interested in biology to begin with. Because yes, science communicators need to do a better job at explaining to the American public what we do, what science does. But I think a lot of the discussion ends up failing when you think of, okay, so how do you do that? Right? Like, it's one thing to be like, oh, science needs to do better, do better, do better. But like, how do we do better? How do we do better when algorithms are what decides what Americans are exposed to, or people are exposed to these days, right? Like, I could, or like an army of science communicators can write news articles day and night, and the average American will never read them. So how do you get your content out to the average American? You have to combine it with entertainment. And so I do that in a certain way, but I am fully aware that the scope by which I do it are for people who already like science. And so we need a generation of people who are under my level, and by level I mean complexity, not under my level as in quality, right? You need there to be high quality information being propagated by young scientists who understand how media of their generation is spread, because I'm making traditional YouTube content and streaming. These are two millennial coded methods of communication, which is only going to work for millennials. If we want a society-wide appreciation for science, I think there have to be content creators that make it sexy to click on. Because I don't want to, I don't want to call you all nerds, but if you are on a Fight the Neutrophil video, and if you are on a Fight the Neutrophil stream, I don't know how to break it to you, but you're probably a nerd. You're probably a nerd. The algorithm determined that you would like my content, and so you are here. We got to get people who don't care about science to care about science. And that's really, really challenging, right? If I were to give a talk about science communication, that's maybe where I would start, right? People blame scientists for not communicating effectively for the failure of the American public to understand what the tax dollars go to. Whereas if you are in any shape or form a little bit invested in science as a thing, it's not that hard to understand what the money is going towards. But the problem is that a lot of America just doesn't care. And we have to meet those Americans where they are, and where they are isn't really where I am. It is for a generation of TikTok dancers to make it appealing for people who don't otherwise care. I think that's really, really important. And I think that in between me and them are probably some of you all who are hearing me talk about this. There are university students amongst you who are more equipped to be on new media than I am. And so, when you are all doing science communication, you who are in the sciences and are currently being educated, you are much more aware of how content spreads than I am. And it is the baton gets passed to you, right? Like, I can only do so much. I am a cell of the algorithm of people who like hearing about diseases already. My hope is that people who hear about a scary disease come in and stay long enough to learn something, right? Like, the average American who cares about scary diseases, they'll find me probably eventually in some form or another if I ever get big, right? But the place that I will not be able to touch with the algorithm are people who don't care about science at all. And there's got to be a way that your generation can figure out how to make it trendy. Oh, sorry, viral. Trendy is what we used to say. I'm not that much older than you are, but I'm old enough where I'm old enough where Vine was a thing, I'll put it that way, and I have not adapted. Even though I have a TikTok channel, I've not posted anything to it in months, I think. The average person only knows the chemical formula for olivine and 1 or 2 feldspars. Common trivia, surely. One thing that I've seen is most normies don't even understand what science is fundamentally, and that is very frustrating, right? Like, there has to be science communication at all levels, right? There has to be a direct connection from the ivory tower to people who are invested. That's one level. And then a connection between people who are like conscious of science in some form or another. So there needs to be that bridge. And then there needs to be a bridge to people who don't care. And that, I think, is the hardest bridge to get over. I feel like TikTok is one of the most misinformation, though, unfortunately. It spreads like wildfire. You'd be 100% good at improving that. Not me. I'm not the one to do that because I'm too old. I'm too out of touch. I'm too out of touch with how short form content goes viral, right? Like, do I have 1 million views on my Neurax worm video? Yes, I do. I'm very proud of that. Brag, 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 right? But the parts of that video that went viral, that's just not what I want to do, honestly. And also, if you look at the comments of that video, you would know why I'm so frustrated that's the short that got 1 million views. But there has to be someone to do that better than I could, right? Like, I'll continue to do the Plague Inc. series at some point. I think it's good. I think it's a nice entry point for people who don't. I think it's not. The people who don't care about biology are not going to watch that video no matter what. But I think it's a nice entry point for people who are passingly aware of disease to get into the more deep stuff, which is why I will continue that series once I'm doing content full time. But yeah, in terms of stopping the spread of misinformation, I mean, we got Mylan in the chat. We know an expert in misinformation right here who knows better than all of us that is dangerous and bad. And good information spreads a lot more slowly than bad information does. But there has to be some sort of code to crack with regards to debunking or pre-bunking that kind of misinformation, right? That is an active field of research. And just like how molecular biology is in its infancy, I shouldn't say infancy. I think right now, relatively, in terms of a science, molecular biology is like maybe young child, maybe like 7 years old. Conspiracy theory research and the spread of misinformation research, I think that, Mylan, feel free to correct me, but I think that is actually in its real infancy. That is a baby science right now, and it is going to mature, and we have to be optimistic that there is going to be a way that we can learn to combat misinformation better, I think. I see so many articles about scientific research which don't actually include a link to the relevant DOI. I mean, I'm guilty of that too. I share the journal and the article title, which will let you get the DOI, but I'm messy about it as well. I think that's a bit of a nerd type concern. Yeah, I think you got it, Marlon. On the other hand, I do think some of the less interested feel belittled by scientists and actually want to be taken seriously and not talked down to in a dancing video. I think I had a kind of interaction like this on my flu video where it was with someone who is vaccine skeptical, and it was hard to not feel emotion from my perspective. But I think in that comments, I could get a sense for this is a person who is genuinely afraid of vaccines, and whatever information ecosystem that they are in has convinced them that vaccines are not worth the risk, when in reality, okay, so there's a fundamental disconnect, right? This is a very interesting topic that I would love to get more in depth into later. I'm not fully equipped to deal with it right now, but there's a fundamental psychological principle by which we analyze risk. I say that no vaccine is 100% free of side effects, and there is a very small percentage of people who will have potentially disastrous side effects from a vaccine. And it is taboo to even say that because the average person will latch onto that and freak out. However, the relative risk of something like COVID is much greater. So if you are comparing number to number, the chance that a COVID infection does something bad to you is higher than the chance of a vaccine doing something bad to you. For me, this is pretty straightforward. Because COVID spreads so rapidly and because it is asymptomatic in a lot of people, because it is so easy to spread, because it has neurotropism, which means it can hide out in your neurons, I think it's a pretty no-brainer. I've had every COVID booster. I've had no side effects other than a painful arm and a fever. And the mechanism by which the mRNA vaccine works is decently well understood. It's just that the mRNA vaccine for COVID is pretty young, so the novelty is there. But again, the mechanism by which mRNA works is pretty well understood. I could explain all of that, but if the contagion of fear takes hold, it becomes very, very difficult to convince someone out of that position. It's still worth doing if you feel that they are coming from a place of genuine concern and not ideological warfare, right? Because someone who is trying to be right is very different than someone who is maybe even sincerely looking for a reason to be wrong. I don't know if that person changed their mind. I hope they did. It's still worth trying. Again, if they are ideologically motivated, that's not even a conversation worth having, because it's just wasting time. When you can be talking to somebody who has genuine concerns that can be convinced otherwise with data. And I think the way that I tried to do that was by posing questions like, what is your personal threshold for risk? Like, how many people in 100,000 people would have to have an adverse reaction to a vaccine where you would feel safe that you would want to take it versus the risk of COVID? And I think having that conversation is nice and important. And I felt like I was very respectful, even though that vaccine hesitancy is so triggering to me because it's an emotional disaster, right? Like, it is coming from the ivory tower. I want to yell and scream, but I can't, because that is, as Mr. Winter, what you're saying, that's belittling, right? And I like, my emotion behind it is genuine and sincere and valid, right? Science communicators have been saying that vaccines have been safe for so long, and there's a lot of disagreement from the public. But you don't win people over by being mad and yelling at them. You have to come up with some way, as my hero Darwin did, to make your position palatable, right? Maybe not even convincing. Maybe they're not convinced after the conversation I had, but maybe the way of thinking that I've described is taking hold. Like, the thought of comparing risk, I think, is not talked about enough in vaccine communication, right? The idea that there are risks to the vaccine, but there are risks getting infected. And yes, you choose to take a vaccine, but the risk of COVID complications is much greater. And then it kind of becomes a no-brainer. But the active action, this is interesting because this is research that I am citing that I have a vague recollection of that came from my undergraduate institution, which is an offshoot of the trolley problem, right? Like the philosophical problem, the trolley problem, where taking an action to move that lever is not psychologically comforting. And in the same way, it is potentially possible that electing to get the COVID vaccine is putting undue psychological pressure and causing what I consider to be a miscalculation of risk that would not normally otherwise be there. Yeah. First thing that I have to learn is not arguing with YECs. I don't actually know what that means. Memetics, yes, memetics. Very baby, yes, exactly. Age measured possibly in weeks based on that relative scale. Yeah, I think so. Like, if molecular biology is 7 years old, I think conspiracy theory and misinformation research literally like newborn, maybe. To go viral with short form content, it usually needs to be the most vibrant and attention grabbing content, which takes away from the experience of watching a detailed video. That's why I think I know where I stand. Like, people, like, we work together, right? That's why this content space is so open. If you want to make short form content that goes viral on TikTok, that gets people interested, right? Because the thing is about getting people to care. If you get people to care, my content will be waiting for them, right? It's a pipeline. They will encounter me when they start getting more and more into learning about these things, if they choose to. And if they stop between me and, I don't know, like Dr. Mike, for example, or Healthcare Triage, for example, who are more on the health side and less on the nitty gritty molecular biology side, I would consider them to be more close to the average biology competencies than my content would be, because my content is demanding. It's demanding in a way where Dr. Mike and Healthcare Triage are not demanding. Like their content isn't easy by any means, but on the scale, I'm talking to you at a university level, and there are high schoolers and middle schoolers in chat who are interested, right? My job is to make the opaque ivory tower stuff interesting and palatable, but that's not necessary for effective science communication. You can also have people taking the, stuff that I've digested and making that into a more fun, attention grabby kind of thing that can compete with all the garbage that's out there. Oh, hey, Fi, there's something interesting on your calendar. Oh my gosh. I'm so sorry. I have to go. Okay, sorry, I have a meeting with V-Theorem. Goodbye, everybody. Oh my gosh, I talked over. Bye. Thank you so much for reminding me. Bye.